不行不行。可以听到吗？This is Monica. I'm going to introduce the general state of the iteration. What are the things? 但是应该是通过是你的。但是如果是你开了我说话，不就听不见？不上次说的吗？上次不是因为你开了我说话听不见，你记得不？我开了。对，你不是你的意思不是咱俩开一个吗？我知道，但是上次是你开的时候，我说话根本听不见，离太远了。不是，我的意思是我这个麦我不开，然后你又不理我。那你让我插啥？好了好了，我没有声音。可以可以。好。嗨，能听得见吗？可以听到吗？其他 OK 吗？
可以听到，可以听到，你能听到我说话不？
可以，没有回音。OK，
行行行，
你现在
那你就用你的电脑共享屏幕吧。
那 OK，
你那个
那一开
你那个 PPT 没有问题哦。然后，开始的话，那个，哎，算了，我过来等你。
我觉得你前面你共享吧，后边我共享，要不然中途我得换。
等一下，我过来过来。
前边你共享，后边我共享，因为我中间我要换 PPT，就有个过程，它会很慢。就是前边不是有一个 Webinar 的那个吗？你用，你这块是你共享，就你这块共享，这块因为不用我们说呀，就是主席在主持，然后完了之后到我的时候，我直接在共享，这样不会乱那个。
我知道啊，但是这两个 PPT 根本就不是一块讲的，
不是一块儿讲的。
就是那个 introduction 的 PPT 可能不太能用。
我觉得共享一下吧，你就共享一下。啊，我也觉得不一定，因为他到时候如果他 welcome 下一个人，那个人就要展示要共享，因为他要做 pre 了嘛，是吧？所以我觉得不一定用得上。
然后我那个的话，让我然后
哦，那你要不要听一下我说的行不？啊，我说的这咋读？Godfreys，Godfreydus，我刚查不出来。Godfreydus， This is Laurie Kang. I'm going to introduce the Journal of Cancer Drug Resistance, and our editor-in-chief is Godfried Peters. And this is the editorial department team. Susan Song is responsible for journal management. Laurie Kang takes charge of special issue construction, interview, and webinar. Blanda Zhang is responsible for manuscript processing. And Bella Zhao is immediately charge of conferences, cooperation, and promotion. If you have any questions, please feel free to contact any of them. They will solve your problems. CDR is an international peer-reviewed open access online journal. And the journal publishes research articles, reviews, case reports, commentary, and letters on aspects of drug resistance and its reversal, including drug design, drug delivery, drug distribution, and ciliary drug resistance. These are the data of the total publication and total article views. Yeah. CDR cooperates with many conferences. This is这咋说呀 ？Which show 为什么CDR cooperate with many conferences 就行了吧？cooperate with many international conferences, and CDR is indexed by this database. Most importantly, in May, CDR is Malay officially indexed by PubMed, and in August, CDR is officially indexed by ESCI. And this is some information of indexing database. And this is an editorial board distribution. 67 editorial board members from 17 countries. They are mainly come from United States, the Netherlands, Italy, United Kingdom, Germany, and China. We can see most of them, the H index, 这咋说 H index 吗？是 对。We can see most of them, the H index is very high. It's very high. It's from 60 to 80. Also, this is a junior editorial board distribution. Mainly, they are from United States, Italy, China, and Japan. And most of them is indexed from 10 to 20. With the help of all the editorial board members and the editorial department team, CDR control the high quality article. So we can say high cited articles published from 2020 to 2022. And this is the homepage of our journal. When we have some important things, it will be updated. And at the bottom of the homepage, there are some data. Also, the data will be updated in time. This is article page. We can see all article downloads information, views information, and also we can download the download article on this page. This also is the article page. Most especially every article has a graphic abstract, which could attract more aspect. CDR do some changes this year. From September 2022, we start to hold online webinars to enhance interaction and exchange among scholars. So if you have some thought about holding the webinar, please contact me, and the editorial team will help you to holding the will help you to prepare the webinar information, 对吧? Also, this year we do lots of interviews. From July to September, we conduct 4 expert interviews. We will continue to conduct interviews with experts to strengthen the cooperation between expert and editorial team. At the same time, spread their thoughts to the world. So if you have some advanced current research or some new published articles, we are happy to help you to promote your articles and your recent research thoughts. This is the official account of CDR. We will promote the new article and the journal information of this official account. Please 关注咋说 attention 吗 ？follow 好。if you have the account, please follow us and we can chat on this social media. Last, the 2020 Best Paper Awards has already been set. 这是一个okay. Okay, lastly, exciting news for you, for all of you. CDR site the Best Paper Award, and this award has already been set and will announce on January and the end of January 2023. And we will launch the 2021 and 2022 Best Paper Awards successively. So if you have suitable paper, please feel free to contact us and submit your manuscript. Okay, bye. Thank you all for your support and the precious time. 还有哪个问题？那怎么聊？我感觉这没啥连贯性。
这些连贯性肯定是你自己加点热词呀，你不可能所有的词儿都自己学出去。这块就是对你刚才说的，你是谁，然后做一个什么关于这种 introduction， 然后我们的编辑团队这没啥。这个，然后这块就是我们公司与以下的这些官方的专业的机构都有合作，然后这充分展示了我们期刊的一个专业性，然后以及在学术上的一个严谨性，然后同时。
突然就开始说我们公司了吗？这个完了之后呢？就比如说这就是介绍完 CDR 的，然后 this is data of publications 说都什么，然后继续。
就直接说 we also cooperated with the following professional institutions which proves the professionalism 就是专业性什么专业性
which proof
证明
proof the
专业性和专业性of CDR in publication， 因为这些都是数据库嘛，它证明我们在出版上的一个专业性。
Which proof the
professionalism of publication CDR， 然后此外的话，我们还和 许多的国际学术会议进行合作，去提升。
We also cooperate with many international conferences.
To make a promotion and 就是做宣传，然后增加期刊的一个曝光，去 expand the exposure of the journal.
Expand
或者说 increase the influence of the journal， 提升期刊的影响。
which useful increase the influence of the journal.
然后now we have indexed by the following databases. Especially in 2022, we achieved great progress. 然后这个就是说5月份被 PMC， 然后8月份被那个ESF 收录。
然后访问。And this is the total distribution. And 什么 from 17 countries. 然后most of them from United States and the Netherlands, Italy, UK, and other country, and other European countries, and they're each indexed mainly from 40 to 60. 是吗？这样吗？ which could show their
这证明了我们we show that our editorial board members is are
professional and 专业和什么？这单词还是 which could show the professional and experienced of our editorial board. 专业和啥呀？
就说就你把这个H出来，就是说such 就这样强大的一个编委团队 ，such strong editorial team give us strong support and make CDR can develop to a further 发展的更好 ，develop better.
Such a strong editorial board members could help CDR achieve a more higher platform. Okay, this is the journal editorial board distribution. It's a junior editorial board distribution. Also, we can see most of them from United States and Italy, China, and other European.
就可以简单的说，然后说他们也在期刊的发展上给予我们很大的支持。可以。
那就是直接就是 this is the editorial, this is the junior editorial board distribution. And they also help a lot for the development of cancer diagnosis. With the effort of all editorial board members and the editorial department team, we strictly control the quality of the article, and we can say they are the high cited article published from 2022 to 2023.
We listed some of the highly cited.
比如说那就是with the help of all editorial board members and the editorial team, we strictly control the quality of the article. We also listed the highly cited articles published from 2020 to 2022.
And 然后接下来就说 in order to make CDR to go further and our editorial department may make some innovations in 2022. The first part is about the homepage.
就是为了让我们
发展
的
的更好，然后给你们做了一些创新吧。然后首先是 homepage， 然后
in order to make CDR more 专业吧。
你自己算吗？40了吗？ 差不多过去了。
嗯，
然后就是在 home page，然后大家可以看到，在首页可以看到期刊的一些最新进展，然后
在
同
上面
时可以看到我们的一些数据，然后这样也展示了我们。
Geographic 和 Geographic Abstract，
嗯，
然后在这儿我们可以看到它的，we can see the data of downloads and views。
然后你说像下载的这些也非常的方便嘛。对，然后这个就是图片的摘要，然后图片摘要这块就是帮助作者去提上文章，去进行文章，其他的文章。然后这个 webinar 这块应该没啥问题，就是说我们这个一年，然后后续我们会继续举办这样的活动，然后感兴趣的专家欢迎去参加。然后这个也没啥，这个就主要是就行。然后这个就是说我们同时。各种各样的去就是社交媒体平台，然后去推广期刊发表的文章以及我们的最新资讯。同时我们也会跟相应的专家一起互动。然后如果你没有这个相应的社交媒体账号的话，也欢迎关注我们。然后同时我们有下面的这些就是邮箱嘛。然后有问题的话可以随时跟我们沟通。然后最就是对，然后一个就是激动的一些消息点，就是我们这块我们设置了这个奖项，然后2020的在这个上发布，然后后续也会推出2021、2022，然后欢迎提到给我们，就感兴趣。
那不成。really we really hope the expert who got the
Good morning, everyone.
Morning, Professor.
Just checking to see if my slides can be shared. Can you see the slides?
Yes, I can see it.
Looks okay?
Yeah, it's fine.
Great, thank you. Are you both in Denmark?
No, we are in China.
You're in China? Very international meeting.
Have you ever been in China?
Oh, yes, several times.
Yeah. Which city?
Mostly in Beijing and Shanghai.
Yeah. Yes, we also have the office in Beijing.
And where are you now, in Shanghai or
We are now in Xi'an office.
Yishan?
Yes. So if you are, if you there is an opportunity, we also welcome you to Xi'an.
Oh, thank you. It's a beautiful place. I know it has some
Yes, yeah.
interesting historical.
Muss ich das auch machen?
Ja, da musst du draufdrücken, jetzt ist die Kamera an.
Okay. Aber ich sehe mich nicht.
Du musst zuerst gehen.
Und jetzt. Ist meine Kamera offen? Nein. Und jetzt muss ich mal schauen, wo die Kamera ist. Siehst du das? Nein, komm, kannst du schon schauen, das ist jetzt kein Problem. Ich sehe nur die Kamera nicht. Da. Was ist da?
Da.
Hm?
Du musst sie doch öffnen. Du musst die Wände da öffnen.
Nein, das ist ja nicht offen. Gut. Ah. Hm? Doch. Jetzt sind sie. Screen Share. Okay. Was muss ich jetzt machen? Screen Share. Und jetzt.
Hm? Also Mute ist man hört ja. Das läuft.
Aber
Ja,
ich sehe. Hello.
Hello.
You hear me?
Yes, I can see your slides, but
Not me?
maybe you do not open your camera.
Yeah. Oh, it's my first Zoom meeting.
It's okay. We can wait a moment.
Oh. Nein. Ja, vielleicht. Ruf mal die Mama schnell an.
Hallo Mama, hier steht eine Kamera.
Dass man mich sieht.
Ja. Ja. Präsentation kann man auch schon sehen.
Hi, Anna.
Hi.
Hi, is everything
Hi,
smooth?
Laurie.
Hello. Now I still can see you. Maybe the camera. You could open your camera.
Yes, I try. I don't know. Doch, das ist schön. It's interestingly for the
Just-
video it's open from my side. It's
It's black.
I mean I'm black. I see. Should I
Yeah.
restart?
Maybe we could wait for wait some minutes for Professor Thomas.
Anna, this is Michael. At the bottom of the screen that you're looking at, there should be
Yes.
something that says mute and something that says stop video.
I see the stop video. It's
Yeah.
on. It's not crossed, and the mute is green. That's a strange thing. It looks like now it's crossed, and now it's open. It's should be going.
Yeah.
But my square is black.
So sometimes it helps just to sign off and then come back on again.
Just redo it. I just redo it.
Redo it. Yeah. For some reason your
Yes.
camera is not clicking.
Thank you.
Maybe you can try to cancel your screen.
Excuse me. I just cancel and come again.
Okay, see you later. Hi, Dr. Chen. Hello. Could you please open your voice? I can see. I can hear you.
Dr. Littman has not joined us yet, it seems like.
Yeah, maybe we could wait a minute. Hi, Dr. Chen.
Yes,
Could you please test your equipment? Is it okay?
test my what?
Test your equipment. Let us say you can share your screen and say it's
Oh,
okay.
okay. I'll try to share my screen and see.
So
Yeah, yeah, everything is great. We can say,
So you can see it, right?
yeah, everything is
Yeah.
great. Thank you. Hi, Thomas.
Hello, can you hear me?
Hi.
Hi, everybody.
Hi, Thomas. Good morning.
Oh, hi, Michael. Hi. Oh, I can see Anna is here also. So we are waiting for Cheetah. Let me send him a message.
do you hear me now?
Yeah.
Yeah, but you still don't see me.
Yeah, we still don't see the screen, your PowerPoint.
Now is, yeah, I see another PowerPoint presentation now.
We assume there's nothing covering your camera on your computer.
Hi, everyone.
Hello, Laurie.
Yeah, could you please open the camera? Hello, Thomas. Can we start now?
Yes, okay, so yeah, I'm still waiting for TitoBot. Sure. So we will start now. And let me share my screen with you. So let me just
New webinar.
Yeah, and then this one. Presentation mode. And that will be this one. Right. Can you see the slide?
Yeah.
The title? Does it appear?
Yeah, we can.
Okay, great. So, hi everybody, and welcome. My name is Thomas Littmann, and I will be your host this afternoon, morning, evening to this very special webinar on ABC transporters in multidrug-resistance. And as said, I really appreciate that everybody could make it, as we have a 12-hour time difference between our morning American and evening Chinese participants, as well as several speakers from, well, Switzerland, Germany, and the UK. So thank you all for taking the time to join us in what I hope will be a very interesting discussion on ABC transporters, their role in drug resistance, and even beyond, as you can see from the list of topics, which I'll try to make up here now. And as you can see, we will have 6 short talks and discussions besides my introductions. And I promised Ms. Laurie Kang, who has kindly organized this meeting, to tell you that Cancer Drug Resistance, that's the journal, will sponsor a $500 Best Speech Award for this webinar. I don't know whether that's a cash prize, Laurie, maybe you will expand, but it means that we have to listen carefully to all 6 talks, because after the presentations, we will vote by Zoom, and at the very end, the winner will be announced and receive an on-screen certificate as well as the prize. But as regards the timing, so it is tentative, it's like 2:05 now, Danish time. We may be done earlier, but we will not go over time. So before I start, do we have any questions? Good. In that case, I have a question, which is, why do we die of cancer? And that's often because it's diagnosed too late, because we have to wait for symptoms to know that something is wrong, and we don't have access to early detection methods, because early detection is what makes the difference if we are to cure cancer. And then there's, of course, the problem of resistance. So as you can see on this slide, which is in German, which our German and Swiss friends will appreciate, because it was, of course, the most thorough I could find. It illustrates the progression of cancer until death. And on the left y-axis, I don't think you can see my pointer, but you can look on the slide. We see the actual number of tumor cells, that's this tumor cell size, and when it reaches 10 to the 12 for the mass of 1 kilogram, the patient dies. To the right, we have the number of cell divisions, and the x-axis that could be like months. So let's assume that we have a person feeling fine, symptom-free, until she discovers a lump in her chest, and it appears to be a breast tumor. Then it's operated by the operation, and perhaps she also receives chemotherapy here in the middle, where you see the blue arrows pointing down. So you see the number of tumor cells drops drastically, but not down to 0. And we might have killed like 99.99% of the tumor cells, but the remaining 0.01% will grow, and it keeps growing even when receiving chemo, which again may kill 99% of the cells, and so on. So we have this log-kill model. And what is worse, if I can get this graph up, you see there is growth of resistant cells, which are either resistant or become resistant to chemotherapy. And these cells really don't care. You can throw almost anything at them. Once they are multi-drug resistant, they keep growing until the patient dies from the tumor load. And today we know that this is the mechanism, the classical MDR mechanism is a matter of membrane transport. And why do we know this? Well, we know it because it's a series of ingenious transport experiments published in 1973. That's almost 50 years ago by the Danish physician Kaldane, who unfortunately died a few years ago. And he proposed this leak and pump model. And it is based on a very simple experiment where he had sensitive and resistant cells. Actually, to begin with, he had nuclei. And when he exposed the nuclei to daunorubicin, we see that the concentration or the accumulation is the same, but not and independent whether you have sensitive or resistant cells. But then if you take whole cells, you can see that the concentration of the compound in the resistant cells is much lower, suggesting some kind of lower permeability to the drug. Then if you add metabolic inhibitors like sodium azide or 2-deoxyglucose, we see that the intracellular concentration goes up in the resistant cells, suggesting energy dependence. And if you add a structural analog of daunorubicin, such as N-acetyl-daunomycin, we also see enhanced accumulation in a competitive way, suggesting that there is a competition for an efflux pathway. And then finally, if we add some unrelated drugs like vimblastin or many other compounds to the cells that are cross-resistant, we see the same effect, again suggesting some kind of competition in the resistant cells. And to explain these observations, Daniel proposed this simple leak and pump model, as you see on the upper right, where the drug enters the cell by passive diffusion. Per definition, is diffusion passive, but before it can reach the nucleus, it is pumped out by an active mechanism. And this figure you see with this drawing is from the Journal of NIH Research, which in 1994 nominated Dan's discovery of multidrug resistance as a milestone in the history of cancer research. And now to some history, because the mechanism that Dane predicted was identified in 1979 by Jack Rawdon and Victor Ling in Toronto, and they named it P-glycoprotein, P for permeability, or PGP. And then in 1986, the gene encoding PGP, MGR1, was cloned by Igor Ronninson and Dr. Foho and colleagues at Michael Gottesman's lab. And I must say, it's an honor to have both Dr. Foho eventually and Michael in this webinar, because if it wasn't for your pioneering work, we wouldn't actually be here. So thanks a lot. Now that the gene for PGP had been cloned, it didn't take long to show that if you transfect sensitive cells with the MDR gene, they become resistant. They acquire the full phenotype of multidrug resistance. And this was so the ultimate proof that PGP can cause MDR. And then once the sequence was known, the gene sequence, the primary and secondary structure of the protein and the proteins in this family could be deducted. And as you can see, I have shown here 3 members of this ABC or ATP binding cassette superfamily of transporters. And in my opinion, these are the three most important ones. So on the top one, you have MRP1, which has an extra transmembrane domain here. And then in the middle, we have P-glycoprotein, ABCP1, which has these two homologous halves, suggesting some evolution from a single half structure by gene duplication. And at the bottom, you see ABCD2, or MXR, as we call it in Dr. Forhus and Susan Bates back in 1998, where we found this protein. It's a half transporter, and it needs to homodimerize to form a functional protein. So each of these transmembrane domains typically consist of 6 hydrophobic alpha helixes, and they associate to form this hydrophobic core that binds to and moves compounds across the membrane. And very efficiently so, as you can see from this live confocal microscopy time-lapse of, on the left hand, S1 colon cancer cells, which are sensitive to mitoxanthron. So this is accumulation of mitoxanthron within minutes in the nuclei of these cells. And if we compare the S1 cells to the SM180 cells, which are resistant to mitoxanthron, we see nothing enters the nucleus, but we see some vesicular accumulation of mitoxanthron. So it's very clear, like night and day, that there is much less accumulation of compound in the resistant cells. Now, one of the burning questions back in the 90s was, what does P-glycoprotein actually do? What is its physiological function? And to answer that, the obvious solution was to generate knockout mice. And this was done by Alfred Schinkel in Piet Borst's lab at the Netherlands Cancer Institute. And the problem with these mice was apparently that they had no phenotype. So they seemed to be quite happy without P-glycoprotein until one day, Alfred got a message from the animal facility that all his knockout mice were dead. And when their caretakers were asked what they had done, they replied, nothing. Well, except that they have sprayed the cages with a pesticide called ivermectin because of mite infestation. And then it appeared that ivermectin was lethal to the knockout mice because they accumulated 100 times more of it in their brains than the wild type. So the problem was solved. PGP is a major part of BBB, or the blood-brain barrier. And it's essential for keeping large hydrophobic drugs out of the brain. And it's also expressed, we know now, in the testes and placenta and the gut and the liver and the kidney, where it mediates excretion of xenobiotic compounds or other substrates. So PGP plays a very important role in pharmacokinetics, as we know today. And actually, it's not only knockout mice that are hypersensitive to ivermectin. Also, many dogs, such as collies like this one, have the MDR1 deletion. And as veterinarians say, white feet don't treat because you risk killing your dog. So we know that ABC transporters are important, but do we know all about them? And well, the answer is no. There's still some questions that we would like to address, and I've just listed some of these. How does transport actually take place? What can explain the extremely broad substrate specificity? How do chemosynthesizers interact with PGP? How is energy coupled to the translocation? And what is the actual structure of P-glycoprotein? Because do we have a high resolution crystal structure? Not yet, as far as I know. And is there more than PGP trendier? And we know there are other proteins, other ABC transporters involved. So I will just show you another slide where I spent a few years, like 20 plus, testing some of the different models for the function of P-glycoprotein. And I will not go into detail to these. Some of them are quite exotic, but I will just show you my favorite model, which is on this slide, where you see first the pumping. This is a classical pumping a conventional pumping a substrate across the membrane bilayer. Then we have the Flipase model, which shows that this hydrophobic drug first intercalates between the lipids in the inner leaflet of the membrane, and then PGP flips it, or rather it's a flopase, because from the inner to the outer membrane, it flops it out to the outer leaflet. And then the vacuum cleaner model, which is a variant of the Flipase model, again, the drug enters the lipid bilayer and then partitions, and PGP acts as a phase separator, removing the drug out of the membrane directly. And in favor of this vacuum cleaner model, there was a very elegant experiment from Raviv et al., again from Michael Gottesman's lab, showing that if you label sensitive cells with ENA, which is a photosensitive probe, together with doxorubicin, you get like even labeling of the membrane. So this is autoradiogram showing this. But if you label resistant cells, all the radioactivity due to the photolabeling, when you incubate together with doxorubicin, is concentrated in one single protein, and that is peglycoprotein. I think that's an extremely elegant experiment. So, yeah, calcium is another favorite of mine when you want to study transport, because it's not fluorescent in the AM ester form, but once it enters the cell, it becomes highly fluorescent, and PGP pumps it out even before it enters the cell, again, suggesting that transport takes place within the cell membrane, which I think Anna Seelig will tell you much more about in a few minutes. Now, let's, in the interest of time, skip forward to the ATPase function, which we can study. And we know that it's very sensitive to detergents, to lipids. It has a high basal activity. Maybe it's working as a chlorase. It can be stimulated by drug. It can be inhibited by drugs. It has these ATP binding motifs. And we can make a whole stamp collection of ATPase profiles characterizing different compounds that can both activate and inhibit the ATPase activity of P-glycoprotein. So we know today that this is the model I got from Suresh Ambudkar, again from Michael's lab, that the catalytic cycle of P-glycoprotein can look like this, where you have a closed conformation and an open conformation, where you can trap the conformation by vanadate, and you can actually study what's going on in each of these steps. I will not go further into this, but just show you the, well, almost last slide, that we have an idea based on homology modeling of how this structural change takes place from this high affinity unformed, and then the drug enters in the middle of the cavity, so the ADP bound low affinity off form where the drug can, a compound, leave the membrane again. Again, thanks to Suresh for these homology models. Then I just have one slide to show from our colleague Richard Callahan, who is not with us today, but he has an important point or several points to make, that it can be hard to publish P-glycoprotein resistant studies because of objections. I think most of these objections can be rebutted, maybe except one, that it's not big data, because I actually think that big data can help us today, as we have single cell omics on many different levels, from the RNA to the protein to the metabolomics, who can actually tell us and give us the whole picture rather than a little bit of the picture. But that's another story for another webinar. So I think I'd better stop now and give the word to Michael. Are you okay with that, Michael? So
yeah, I'm fine.
perspective?
I'm trying to get my slides up.
Yes. I'll mute myself then.
It says you have to unshare. Thomas, can you unshare?
To exit.
Says the yeah, it says I can't share until you unshare.
Okay, let me try again. Sorry.
Okay, should be good. Okay. Okay.
Great.
So thank you for that great introduction. And my talk is going to focus on a zebrafish model to study the role of ABC transporters at the blood-brain barrier. You mentioned this as one of the roles of these ATP transporters. This work was done mostly with the help of a postdoctoral fellow in my lab, Joanna Thomas, and Rob Roby, a staff scientist. So very quickly, this is what the blood-brain barrier looks like. You showed a similar schematic. There are endothelial cells, there are parasites, there are glial cells, astrocytes. The barrier is maintained physically by the tight junctions between the endothelial cells, and chemically by a variety of pumps. And you've already mentioned ABCC4, MRP4, but the major ones that have to do with keeping drugs out of the brain are G2 and PGP. Now, these two seem to work together. If you look, for example, at the failure of certain drugs to enter the brain during chemotherapy, you find that knockout mice, these are the ones that Al Schinkel, Anton Burns, and Pete Borst prepared, knockout mice for PGP or ABCG2 each show some increase in accumulation of various drugs, and these drugs are shown here. But if you knock out both the MDR1 and the ABCG2 transporter, you get a big increase in up to 100-fold increase in drug accumulation in the brain, as was seen, for example, with ivermectin. Now, we wanted to have a model that we could use to study the blood-brain barrier and the role of ABC transporters. And Matt Hall and Josh Bakshishian in my lab several years ago created a mouse model in which the glial cells in the brain express luciferase under control of a GFAP promoter. It turns out that luciferin, which is a substrate for luciferase, can't get across the blood-brain barrier because it's a substrate for ABCG2. And the only way to get luciferin into the brain and to generate light in the brains of these mice is to inhibit ACG2 or in some other way abrogate the blood-brain barrier. And we showed that inhibitors of ABCG2, such as KO143, could dramatically increase the light in the brain. Luciferin is able to cross the blood-brain barrier, and luciferase turns it into light. Now, this is a nice model. It's quantitative. It's a good way of measuring disruption of the blood-brain barrier, but it's expensive because it's mice and doesn't allow for high throughput analysis of agents that might be disrupting the blood-brain barrier. So we decided to go ahead and develop a zebrafish model for the blood-brain barrier. And we were interested in sort of three major questions. What, if any, are the zebrafish homologs for ABCB1 and ABCG2? In other words, can we clone them and identify them and demonstrate that they are expressed in the zebrafish endothelial cells in their brains? Do these proteins, if they exist, have homologous substrates to the human ones so that they would be useful for pharmacologic studies? And of course, which homologs are expressed at the zebrafish blood-brain barrier? Now, the first step was to look at the genetics of zebrafish ABC transporters. And it turns out that for the human ABCB1, there are two homologs in zebrafish, initially called ABC1A and 1B. They were later renamed, for reasons I do not understand, ABCB4 and ABCB5. Of those two, ABCB4 is the most homologous to human ABCB1 with 63% amino acid identity. And it also is syntenic. It seems to, it's on chromosome 8 in, it's on chromosome 16 in Danio Ririo, which is the zebrafish. But it is associated with similar genes to ABCB1 and B4 in the human. So it's syntenic and it's highly homologous. So the next step is it expressed in the brain and brain endothelial cells? And this is a study using several different techniques to visualize expression of proteins in endothelial cells in the brain. On the far left, you see a marker of capillary endothelial cells, FLE1, GFP labeled, and you can see these sort of capillary-like structures. C219 is a monoclonal antibody developed in Vic Ling's lab, which identifies all forms of P-glycoprotein, which would be ABCB4 or ABCB5 in the zebrafish. And you can see similar localization. And when you look at both together, it's interesting, they're both in endothelial cells, but they're in different parts of the endothelial cells in the brain. So the question was, which is this ABCB4 or ABCB5? And so we created models with transgenic, with cell lines that expressed zebrafish ABCB4 or B5, or MDR19 represents human PGP. And you can see C219 recognizes all of the cells. The riboprobes for ABCB4 only recognize B4. The ones for B5 only recognize B5. And when you go back into the brain, in the upper left, You can see C219 identifies, as we showed before, endothelial cells. On the upper right, ABCB5 shows no localization, but ABCB4 shows good localization. These little yellow dots that you see represent ABCB4 RNA being expressed in the endothelial cells in the brain, where we see P-glycoprotein, which is ABCB4 protein. So what is the substrate specificity of these transporters? This shows sort of typical killing curves. MDR19, again, is the human P-glycoprotein. And you can see if you look at these fairly typical anti-cancer drugs, etoposide, paclitaxel, vinblastine, doxorubicin, that ABCB4 confers resistance in cells to all four of these drugs, but that, for example, for doxorubicin, ABCB5 doesn't. So B4 is very much like ABCB4, and the zebrafish is very much like human ABCB1 in terms of substrate specificity. And Matt Hall, a colleague of ours who does high throughput analysis of drug resistance, was able to demonstrate, as seen in this slide, that using about 100 different substrates, that the correlation between the ability to confer resistance for the zebrafish ABCB4 was much better compared to ABCB1 than ABCB5. You can see the data here showing very clearly that B4 is much more like B5. Incidentally, Disha in our lab has recently done some structural analysis of ABCB4 in the zebrafish using cryo-EM studies to pretty high resolution. And it looks, as far as we can see, it looks identical to human P-glycoprotein in terms of structure. ABCG2, we also did the genetic analysis. Turns out that there are 4 homologs of ABCG2 in the zebrafish, A, B, C, and D. None of them are syntenic, but ABC2A is the most homologous with 60% amino acid identity. And again, when you look at the substrate specificity, ABCG2A looks much more like human ABCG2 than ABC, B, C, or D. We used a riboprobe against 2A and again demonstrated that it was expressed in the brain of the zebrafish. So let's look at the zebrafish model. And this is basically what the blood-brain barrier looks like in the zebrafish. And we decided in generating the zebrafish model to not use luciferase, but to use an engineered luciferase called Nano Luciferase from a different source. This is gives a more light signal. We thought, given the small size of the zebrafish and the complications of administering the various drugs, that we would be better off with a higher yield of light. And it has a different set of substrates, mostly calantrazines. When we looked at the ability of a variety of different nanolook substrates to activate the nanoluc in various cell lines that have been engineered expressing different ABC transporters, we got the following results. It's a little complicated, but let's look at the graph on the left. Furamazine is a substrate for nanoluc. When you add them together in a test tube, you get a lot of light. The quantum yield is very high, and you can see on the ordinate that there are lots of light units coming out. The first graph shows the normal zebrafish in which the furamazine is unable to cross the blood-brain barrier, and there's no effect of inhibitors of ABCG2. And then you can see various different transfected cell lines with human G2, ABCG2A, human P-glycoprotein, and ABCB4. And it looks very clearly that if you had an inhibitor to ABCG2, you get a big increase in light in the for human G2 and G2A, but you don't see an increase for PGP and B4. That means that furamazine is a substrate for ABCG2 and ABCG2A in the zebrafish, but not for PGP. Colentrazine in the middle graph appears to be a substrate for both, and colentrazine H is a substrate for G2A and maybe for ABCB4 in the zebrafish. And that's summarized on the bottom here. What it means is we have a variety of different substrates that are either G2 or PGP, transported by G2 or PGP, that can tell us whether the material is getting into the zebrafish brain. And this shows some of the data in actually living zebrafish. Put the zebrafish in a little bit of water, you add the drug, you add the inhibitor, you add the drug, and then you measure the formation of light. And on the left, you can see it's time dependent. It's dependent on the presence of inhibitor. And the inhibitor that we use is a standard KO143 inhibitor of ABCG2. So as a result, we now have a really lovely assay, which is quantitative, which allows us to measure the intactness of the blood-brain barrier in zebrafish in a quantitative way. And the intent is to look for interventions that can inhibit any of the ABC transporters or actually cause other disruptions or increase transcytosis to get materials into the brain. So conclusions, ABCB4 and 2A function as efflux transporters at the zebrafish blood-brain barrier. B4 and G2 functionally phenocopy human PGP. There are several potential substrates for these transporters that can be used for our studies. Furimazine, which is an ABC2A substrate, gives the best signal and can be used in actually intact animals to demonstrate the formation of light with inhibition of the blood-brain barrier. And finally, as I said, the assay can be used to measure in a quantitative way the effect of any agent that disrupts the blood-brain barrier and allows furimazine to enter the brain. And I want to thank my laboratory and the many people who've contributed to our studies over the years. And I'll end there and hopefully am on time. Thanks a lot, Thomas.
So thanks a lot, Michael, for a very interesting and illustrative and exciting presentation on zebrafish and the blood-brain barrier. So good question. Do you envisage that this could become an assay that the FDA would like us to use for testing drug interactions?
Yeah, we think it has a lot of potential. It's quite easy once you know how to grow a zebrafish. And I have to say, I remember as a child having zebrafish in my tropical fish tank. So I think it's pretty easy. It should be an easy assay. And you need some way to measure light, and light can be quantitative.
Excellent. I can see Ian has a question. You have raised your hand.
Yeah, thanks, Thomas. Thanks, Michael. That was a really interesting talk. And actually, your data on furimazine explains why we couldn't get a nano-Brett assay for ABC to work. But what I actually want is what
Can't you?
Ian, I missed that point. So you were trying to use it for PGP?
No. We were trying to set up a nano-Brett assay with ABCG2, and your data on furimazine might explain why that assay was problematic for us. But actually, what I wanted to ask you was, what do you think the function of these homologous family transporters?
Ian, you're breaking up.
Yeah.
Will I be best to type it in the chat? Okay.
Put in the chat.
I'll put it in the chat.
Actually, there was one more question, but let's have Ian write his, because Michael, a question from Zilukenbach. Did you investigate the transporters in the brain of adult zebrafish? And do you have an idea from which developmental stage of the transporters are expressed in the brain?
Yeah. Yeah. So that's a great question. And that was one of the questions we wanted to ask. The studies that I showed you were done in 6 DPF, 6 days post-fertilization zebrafish. So basically in early larval form, relatively early larval forms. We are about to try to get this to work in adult zebrafish. One of the interesting issues in adult zebrafish is that some of these ABC transporters are expressed at very high levels in the skin of the zebrafish, ABCB5, for example, which is a pretty good drug transporter. So it may be harder to get some of the drugs we're interested, the inhibitors and so on, into the adult zebrafish, but we're about to start working with adult zebrafish. We've looked at both expression of ABC transporters and the intactness of the blood-brain barrier at different stages after fertilization. At 3 days, we see a clear signal. The baseline is somewhat higher. We need to repeat these experiments, but so it looks like it might be a leaky blood-brain barrier, and there is both G2 and ABCB4 expressed at 3 days post-fertilization. So there is an intact blood-brain barrier even 3 days after fertilization, and then it increases in capacity and tightness as the zebrafish matures.
So now I've got Ian's question. So it says, Michael, your ABCB5 and other G2 homologs, any thoughts as to their roles, other sanctuary sites, or just no MDR pumps at all?
Yeah, so when we did the immunohistochemistry for the example, the ABCB4 and B5, you can use C219 and you can do in situ probes. The distribution is really quite interesting. So B5, as I mentioned, is expressed, for example, in the skin, in the gills at very high levels. So you could imagine that one of its functions may be to protect the animal against toxic materials in the water in which they're swimming. The ABCG2, we don't have the complete studies yet, but each of those has, we're pretty sure, other functions. And we're sort of sorting through. Each of those is a different project in its own right, but that's a great question, and they're going to turn out to be interesting, I'm sure.
Okay, so thanks a lot for questions and answers, Michael. Nevertheless, I would ask now Dr. Chen to prepare the next talk.
First, I just want to make sure that you could see my slides.
Maybe you would like to go to presentation mode. Otherwise, we will have to read your comments.
You still cannot see?
We can see, but it's in your edit mode. It's not in presentation mode.
Oh, I see.
Now, well, yeah, that one. Yeah, try again, the presentation at the bottom. Yeah, that one.
Didn't work.
Maybe it's because you are sharing your edit screen and not your presentation screen.
Okay.
Well, you can also maximize that window. Then you can just scroll down. If you maximize the window, you can see in front of you. That might work.
So let me share again.
Yes, that's good.
So whenever I got to this place, I could not get.
Yeah, try to click. Translators.
Yeah. has stopped as the share window is closed. Okay. Let me see share again. No.
Now we are in your file browser.
Thomas, it wasn't all that bad the way he had it. One could see it.
Oh,
If
hi,
you can
Tito, is it
just-
you? 844264. Hi.
Hey, how are you? If you could just eliminate the left side of it,
Yeah.
just move
Okay,
it
so-
over. That's what I'm gonna have to do with mine. When I go to presentation mode, it doesn't advance.
Ah, that's it. Okay, so go back to the first screen you showed us, your edit mode, and then just maximize it.
Yeah, lower the bottom part so we don't see his comments, move it over to
Yeah.
the left so we don't see the all the slides in small. And that usually works well enough.
Yeah. So if you just click now on your final CDR, that should open. Or would you prefer that Dr. Anna Seelig gives her presentation first so you can fix your PowerPoint? I can't hear you. You are muted now. Jen, you are muted. Right. So, Dr. Chen, you can prepare and do it later. Is that okay with you? Because then we'll just take Anna Seelig's presentation. Anna, if you are ready, you are also muted.
Still muted?
No, it's Anna Seelig who is muted. Now you are on mute.
You hear me?
Yes.
I can hear you.
Because, yes,
Good.
because then we can take Dr. Seelig's presentation first and take yours
Yes,
later.
yes.
When,
Okay, go
okay,
ahead.
let's do that then. So Anna,
Thomas,
it's all
I do-
yours. Hmm? Yes.
Share screen, huh?
Do you want me to share your presentation that you sent? Anna, do you want to share your own screen? Oh. So.
So I should start, Thomas?
Yes, please.
Okay, good.
And do you have your own presentation or do you want me to share it from my computer, the one that you sent?
No, I have my own one.
Oh,
Do you see
good.
it?
No, I see you.
You see me. So
Mhm.
I have to get myself out. So I see the.
You have the share screen button in the green button, share screen.
Yes, I did this actually.
And then it will probably ask you which screen to share.
Share screen.
Yes.
No. You see it
Yes.
now?
Yes. Now we
Okay,
see
very
all
good.
your slides. Then
Good.
just
Now
start
I
with
have presentation mode.
Mhm.
Okay, now I have just to get
Yeah, it works.
the beginning. So, okay,
Yes,
good.
excellent. So it's all yours. Take it away, Anna.
Okay, so I would like to talk about the forgotten thermodynamics and kinetics, and I did a review of these lot of publications, about 24 years of work, and at the end I found a new, or I would like to present a new model for the function of ABCB1. Now, we are all under the spell of these wonderful structures, and we kind of forgot about the physical chemistry and the kinetics. And here we saw these nice structures and constructed a model. And it's actually Kenny Linton and Higgins who first proposed this model. They saw in their crystal structures, faint crystal structures since at the beginning, they saw these A-shape and V-shape transporters, and they said, that's a switch. We have no ATP bound. We have an open conformation towards the cytosol, and we bind then the molecule goes in through the aqueous channel, and we have a switch, and the molecule can leave to the outside. And these are the structures of P-glycoprotein and of SAB, and these structure kind of confirmed this model, or seemed to confirm this model. And my question was then, where is the membrane, and what is the role of the membrane here in the middle of this transmembrane helices? What does it do? And we know that the first publication of P-glycoprotein was by Giuliano and Ling, and he identified the transporter, and he found that the substrates were all amphipathic or amphiphilic. And there are more things to remember. We have a physiology. We know that the ATP concentration is pretty high in cells. We have homeostasis. We know that the Km value is pretty low, which suggests that we have practically always in the resting state 2 molecules of ATP bound. So this structure, which assumes open conformation with no ATP bound, is for me difficult to understand. Then we have a second point, the nature of the membrane. It determines the interactions of the amphiphilic molecules which penetrate into the membrane. And there is a lot of work by Michael Gottesman group's early work, who showed that the allocrides bind through the membrane to the transporter. They are the membrane is hydrophobic. It's anisotropic, which means it is structured. It's fluid, but it's still, there is a structure. It has a certain packing density of these lipids. And very importantly, it has a dielectric constant. So the dielectric constant of the membrane is very low. Then we have kinetic factors, and we will see whether these, we had in the very early PGP titrations were all biphasic. And with time, the inhibitory phase disappeared. And we will now see. So, and we will see whether this is real or not. Now, the membrane has leads to a strong accumulation of the substrates. We have here a simple model of the transporter. The molecule is bound, it is exported, and in parallel, we have passive flux. We have substrates which are amphiphilic. I showed the molecule in the way I would sit at the air-water interface. Air is hydrophobic, like a lipid membrane, and water is, of course, hydrophilic. So you see these encircled groups, which are hydrogen bond acceptor groups. And we found certain patterns. The first one is colchicine. And colchicine, you see, has a pattern with 3 atoms in it. And we proposed quite a time ago that these groups are inducing P-glycoprotein. Then the second one is a cancer drug, which is very hydrophobic, very small. It penetrates very much into membranes. And the third one is rhodamine. And this is a related compound. And the difference in the C and D molecule is that we have 2 patterns in rhodamine, but we have only 1 small pattern in the analog. And the reason is that the analog has a NH2 group instead of a tertiary amino group. And these NH2 groups do not form patterns. So patterns are only formed by tertiary amino groups, tertiary or quaternary amino groups, which have a positive charge. Now, this is a very early slide, which just shows the distances between the patterns. So we have 2 types of patterns, a type 1 pattern with 2.5 angstrom and a type 4 pattern, the induced pattern with 4.6 angstroms. What do these patterns, what are they good for? So here is a transmembrane part of the P-glycoprotein. And you see very many hydrogen bond acceptor groups in these transmembrane helices. And surprisingly, this always intrigued me. They look very much to the outside, not so much to the small channel. And so the role of these groups here, do you see my cursor? Yes. The role of these groups is to attract the hydrogen bond acceptor groups of the substrates from the membrane. Now, the possible interactions are hydrophobic interactions, electrostatic interactions, and they work if the molecule is in the aqueous phase and binds to the membrane. Then within the membrane, we cannot attract a molecule by hydrophobic interactions because the most hydrophobic medium in a cell is the membrane itself. So you cannot take it out by hydrophobic interactions. However, you can extract it by weak dielectric interactions, like cation P interactions. That's the most efficient one. We measured 6.5 angstroms, 6.5 kilojoules per mole for this interaction. The hydrogen bonding interactions are 2.5 to 4 kilojoules per mole. So people who have read early literature about hydrogen bonding would think this is a little bit low because one said 10 kilojoules or more. These are the calculated values, but I found recent literature, not very much, but I found some references where they measured hydrogen bonds in real membranes in a protein, and they found the same range as we found for the interaction between the allocrite and the transporter. Then we have pi-pi stacking interactions. That's the phenyl group, which slide over each other. And we have, of course, van der Waals forces. The two red ones are the most important ones because p-cation interaction, a very highly charged molecule will not penetrate the membrane. So this is more for theoretical interest than for practical interest. And then the next point is the kinetics. We measured ATP hydrolysis by the classical method in inside-out vesicles, but also in cells. And for this purpose, we used the NIH cells. We are very grateful to Michael Gottesman that he has given us these cells, and we measured the ATPase activity as a function of concentration. Now, if you have low concentrations, we have the activation. If we go to high concentrations, we have inhibition. And this shape was discussed a lot less in papers actually as in small talk, I would say. And the second part here was a bit discussed. So is it aggregation which leads to this decrease, or is it is it too little ATP available? Is it a disturbance of the membranes? And we tested all these individual facts by physical chemical techniques. We did for the disordering, we did deuterium NMR. For the aggregation, we did light scattering. And for the exhaustion of ATP, we did ATP regenerating assay. So we are very sure that the down regulation is real, and we could not explain the novel PGP inhibitors without having two molecules bound to the transporter. And actually, it's now also proven by crystallography that two molecules can bind. So this is a very important factor. And then is an interesting experiment. We said that the membrane shapes the interactions. Now, the diffusion into the membrane decreases when you have a high packing density, because you have to invest more energy, or the molecule has to invest more energy to penetrate between the lipids. On the other hand, the binding directly within the membrane, the binding of the allocrite to the transporter increases if you have a higher packing density. And this is because the binding, the weak electrostatic binding, is very much dependent on the dielectric constant of the membrane. And if it is very low, the affinity is high. If you have high affinity, the rate is low. So we have on the left side, we have a membrane. We have the transporter in the membrane, and we have a relatively low rate. Then we put the transporter in a mice cell, and we get a very strong increase, almost threefold increase in rate. And this can be explained by the dielectric constant change. You can quantitatively calculate this effect. And interestingly, Suresh Ambudkar has done quite a big analysis of ATP hydrolysis in a very recent one in vesicles and in cells, and he found a very similar threefold increase when he goes to mice cells. And this was particularly interesting because he did it for these novel inhibitors which bind as dimers in cells. When he put them in mice cells, he got activation. So he had a switch from inhibition in cells to activation in vesicles. Now, we come to our suggestion. So we summarize here. First, we have the binding step consists in two individual steps. First, we have hydrophobic interactions. So we have penetration of the molecules based on hydrophobic on the hydrophobic effect into the membrane. In the cytosolic leaflet, we have in addition electrostatic interactions, which are, however, not very pronounced because we have a lot of salt in the cell, magnesium, which shields the surface potential. And so this is the first step, hydrophobic step. The second step is these dipolar interactions in the very low dielectric environment. In the middle, it's about epsilon equals 2. In the interface, it's about 30 to 40. And in the aqueous phase, it's 80. So from here, from the aqueous phase to the middle of the membrane, we have a factor of 40 increase in binding or in attraction. And the blue part here reflects the polar groups, and the yellow part, the hydrophobic part of the molecule. And our suggestion is that these molecules are attracted to the surface of the protein. We have two molecules. I should say we also did molecular dynamics simulations, and we found that the molecule closes. We have always these open structures because we have no ATP when we do a crystal structures. If we add the ATP, we have a closing of the structure. So our starting point is this resting molecule with two ATPs bound. The allocrides bind to the transporter. At some point, it probably gives a distortion a little bit, and we have ATP hydrolysis. So this is the next slide. So we have this first binding step here. This induces ATP hydrolysis. We have phosphate release. The molecule sitting here, this means just that we have this binding process. Oops. And this distortion leads to an opening of the transporter. Water flows in, and due to the income of water, the molecule immediately turns around such that the hydrophobic part goes to the aqueous phase, and the hydrophilic blue part goes to the aqueous phase, and the hydrophobic part remains in the membrane phase. Then we have, this is ADP, ATP, these two states here. Then we have ATP rebinding. The molecule closes, and if there is something left, it's squeezed out into the lipid phase. So this is maybe surprising if you are used to the alternating excess model, but Camilo Perez and Lojo also observed a similar or suggested a similar approach or a similar model for a glycolipid. And they were very puzzled at the time, and interestingly, they did this structure with two ATP analogs bound. So it was in principle this structure here which they presented in their X-ray structure. And so we looked at the kinetics of very many four transporters. So the P-glycoprotein, which was a gift, as I said already, from Michael Gottesman. We had CFTR, which works in a very similar way. Then SAF is an analog of P-glycoprotein, also with interleaved helices. And then we have BCRP. BCRP takes non-amphiphilic compounds, whereas PGP takes the amphiphilic compounds. And but the interaction is very similar, actually, in P-glycoprotein, CFTR, and SAF. We have amphiphilic molecules which probably bind in a similar manner. And yeah, that's so much for my presentation.
So thank you very much, Anna, for this very interesting presentation on the mechanism of action of P-glycoprotein. Actually, I think it's still compatible with the hydrophobic vacuum cleaner. It's more in the detail of what's happening.
I think the hydrophobic, the hydrophobic attraction is certainly there, but it's not to the protein, it's to the membrane. And once in the membrane, we have these weak electrostatic interactions. So the membrane is actually a hydrophobic vacuum cleaner, if you want. So I would call it an electric vacuum cleaner or electrostatic vacuum cleaner rather than a hydrophobic one. But of course, the hydrophobic effect, the first effect is still very, very important.
Okay, I've got one question in the interest of time from Ian. I'm reading up interesting work on it. It ties with some biochemical data we have from mutants of ABCD2 and ABCB1 with Rick Callahan, that there are superficial interactions between the drug and the protein surface via the lipid bilayer. I ask you to speculate as to why the EM and crystal structures pick up cavity-bound rather than surface-bound substrates? Affinity, lack of membrane in the structural studies? Maybe Ian will explain. Yeah, did you get the question?
The cavity bound, I mean, the point, are you thinking about structures?
Yes. I think this is Ian's question. Yeah.
I'm not, this is a difficult question. I mean, what I'm sure, you can never be sure. What we saw in our molecular dynamics simulation was, and we also analyzed a lot of deer data, and we don't find unambiguous behavior of the, is it closed, is it not closed? So it's open from point of view of deer. And the question, if I got it correctly, is does it go into the cavity of the small cavity of the transporter? Is that correct?
Well, it's rather whether the crystal structure pick up cavity rather than surface substance. But I think Michael Gottesman has a comment or a question.
Yeah,
Michael?
I do. Thanks a lot, Anna. That was great. I want, if I understand correctly, at the end of your second step, the substrate is sticking into the outer leaflet of the plasma membrane with the hydrophilic part coming out. And I'm going to suggest the third step. There's a lot of data, old data that no one's ever followed up, showing that the transport process is much more efficient in the presence of albumin in the extracellular space. So the third step may well be that you need an acceptor.
The extraction of the
extraction
I completely agree. We actually measured drug binding to albumin, and it's very much hydrophobicity or amphiphilicity is a big factor for albumin binding, and this is a very good point. I think Al-Shabi measured, you may remember, he spin labeled verapamil and measured, he wanted to measure how much gets out in the aqueous phase, and he had a quaternary ammonium ion on verapamil and thought it's so charged it must go into the aqueous phase. But it remained to 70% in the membrane and only the charge was sticking out. So I think it really needs something to extract it from the membrane. And there is another point, this CYP, of course, also works in the membrane, makes it more hydrophilic, and then it will leave the membrane also. So there are many factors working on it.
I just wanted to defend the term hydrophobic vacuum cleaner, which I coined. The intent was that it was a vacuum cleaner that removed compounds from the hydrophobic bilayer. It itself was the vacuum cleaner.
Okay.
So I think that's consistent with your model.
Yeah. It's I just the point which when I did this review was kind of, I mean, I always propose this electrostatic interactions, but the point that when I looked at the numbers, that the epsilon, the dielectric constant, makes such a big effect in the affinity, and that you see this then afterwards in the rate, because as do you see this? Oops, no. Do you see the slide with the 3 transporters here? And so if the affinity here, it's a bigger molecule which has a lower weight, but the bigger molecules usually have a higher affinity. Now, if you think of a reduced affinity just because of the lower dielectric constant of the micellar environment, you have this lower affinity and you have a higher rate. And this is, I mean, the Suresh experiment and our early experiment with in SAF. So this, I only calculate the numbers now, and this was, yeah, something which brought me to this model, actually.
I see Till Luckenbach has raised his hand.
Yeah, actually, I'd like also to ask you something. Thanks a lot for this talk, Anna. So when you say, actually you don't say it, but the others say vacuum hydrophobic vacuum cleaner, which but actually to be actively transporting or to be effective as a transporter, a compound cannot be fully lipophilic because it diffuses too fast through the membrane. So it needs to diffuse slowly through the membrane to be able to be actively transported. And I was just wondering, you know, so you need a compound that has hydrophilic and lipophilic properties in a way. And I was wondering, do you know how fast these compounds that you have as substrates or recognize as substrates, how fast they actually diffuse through a membrane?
Yes, if you see this slide here, you see the green, actually here on the y-axis is the passive influx, calculated passive influx. These stars here with the line is the transport activity. And on the x-axis, I have the cross-sectional area of the molecules. Now, passive diffusion is extremely dependent on the packing density of the membrane, on the size of the molecule. So it's an exponential dependence on the size of the molecule. And you have you see the molecules here, the small ones up here, they are extremely fast. So, oops, they are extremely fast and they go through. So green is for green light, you can walk, they just go. And the red ones have to stop because they are too big. Their passive flux is in the order or even lower than the active transport rate of the transporter. So the stars here are measured values for transport. And interestingly, the transport is much less packing density dependence. This may be strange for you when I talked about the micelle, but micelle is really something very different. The dielectric constant is very different in a micelle. And for the transporter, we have a fact, we have very little difference because the penetration in the membrane is lower when you have a high packing density, but the affinity to the transporter is higher. So we have a kind of compensatory mechanism with respect to the packing density, which is not the case in the diffusion. There we have the just strong dependence, exponential dependence on size and packing density. So you can, in principle, roughly estimate if you calculate passive diffusion. Actually, it's even easier. You can say all molecules which are larger than 70 to 80 square angstroms in diameter, they will not. Maybe I have one other slide. Yeah, this one. This is how I entered the field. We measured, this was together with Merck in Darmstadt. They wanted to know whether one can predict CNS compounds which penetrate into the brain or compounds which do not penetrate into the brain. They wanted to make antihistaminics which do not penetrate the brain. So we measure the air-water partitioning. So how well does it go to the air-water interface? And we measured the CMC of the compound, and we measured the cross-sectional area of the molecule also by surface activity measurements. And we found that the small compounds lower than 80 square angstroms, around 70 square angstroms, they penetrate the brain, and the larger compounds do not penetrate the brain. And I gave a talk at a meeting, and someone, I'm a membrane person from my start. And when I started out and I measured this, I calculated passive diffusion, and the person asked me, what about P-glycoprotein? Could it have an effect? And I was my most difficult question ever, because I had no idea about P-glycoprotein, and I was very much worried. I went home and immediately searched for P-glycoprotein. At that time, we had no Google, so it was a bit more work. And this is how I came to the P-glycoprotein to work on this interesting molecule.
Well,
So
I'm glad you found it, Anna. And thank you so much for a very interesting both talk and discussion. In the interest of time, we are running a little bit late, half an hour or so. I think we should ask Dr. Chen to put on his talk if it's ready.
Can first
I have to get out
and share, and so I can share.
Stop share. Yeah. Okay, good.
Yes. Looks good.
You can see the slides?
Yes.
Okay.
Slide.
So I'll start, and also I promise I'll keep it short so that I can save some time. And thank you very much for your patience. First, I would like to thank the Journal of Cancer Drug Resistance and Dr. Lippman for organizing this meeting and inviting me to present in the study we have done in the area of extracellular ATP induced and ABC transporter-mediated drug resistance in human lung cancers. And first, I have to admit that unlike other participants, and I'm not an expert in ABC at all. Actually, it is our cancer study with extracellular ATP that lead us to this area. Hopefully, I can share some data and you'll find it interesting. Can you see the, so you don't see the move of slides?
No, it's still on the first slide.
Yeah, so somehow, that happened.
See, that's what happens to me, Thomas. I put it,
Yeah.
sometimes you put it onto, you know, this presentation mode and then it won't advance. And he might see it advancing, but we don't see it advancing, which is why better to just go to the,
Right.
you know, just maximize your screen.
Yeah, you might want to do that, Dr. Chen. As the
do what?
Cheeto suggested. Just go back in edit mode and maximize your screen and just show the slides from your edit screen window.
Now see if I could do that. Hold on. But if I go to this my PowerPoint, it still doesn't move. So what can I do? So my original file, if I open,
Hi, Dr. Chen.
yes.
Maybe you can send your slides to my email, and I can try to share the stream on your behalf and see whether it works.
Okay. So Dr. Littman, please go ahead to let the next speaker to speak. I'll send it my PowerPoint.
Till, Dr. Till Luckenbach, would you be ready to present your slides?
Say it again.
It's I'm addressed Till. Yeah, are you okay?
Yeah, we can do it.
But then Dr. Chen has to unshare his screen, I guess.
Yeah, I'll unshare.
Unshare or stop sharing. Right.
Okay, you see? Great. All right. I also talk about active transport in zebrafish, which we heard already a bit today from Michael Gottesman. And I'll talk about zebrafish embryos. And I'm at the Helmholtz Center for Environmental Research, so I'm not really in medical research. It's really more environmental research. That's the background of my work or our work. So we're interested in ecotoxicological effects of environmental chemicals and how these effects can be used to assess environmental risks. And well, actually, you're actually required to do for tests of chemicals with adult fish, but these are animal experiments, and this raises the questions: why not use fish embryos as an alternative? Because they are not considered as animal experiments. They are faster, cheaper, and there are lots of diagnostic options in the embryo. But well, you need data for adult fish, actually, for chemical registration. And when you compare the toxicity of chemicals to embryos and adult fish, you have a correlation, but you have a lot of outliers also. So it's a question if you can make you know, a good, let's say, prediction of toxicity for adult fish from fish embryos. And therefore is the question, how can fish embryo toxicity really serve to predict adult fish? So we need to understand why chemicals are toxic to adult fish and why they are toxic to fish embryos, so we can kind of make a prediction for adult fish, basically to not do so many animal experiments. And as you can see, I mean, fish and adult fish and fish embryos are quite different. You can see it already here. And the embryo is a very dynamic system. So this is a zebrafish embryo. Within 3 days, it develops from a single cell to a complete organism. And it doesn't take up any food. So during development, so in these 3-4 days, first 10 days, it actually doesn't eat. So it loses a lot of weight. Body mass parameters decrease. So you see here dry weight, carbon, nitrogen. And also gene expression is extremely dynamic. So this is transporter protein that we heard already today about, and which I will also talk about. It's zebrafish ABCB4. And you can see that is the constitutive expression of this transporter in the first, let's say, 5 or 6 days, I think. And these data are actually from omics data, so microarray and sequencing data. And Paul Michaelis, who works in our lab, he kind of analyzed the control data from experiments, the control data to look for constitutive expression of this transporter gene. So basically, we try to understand the parameters that determine toxicokinetics. So it's equivalent to pharmacokinetics of chemicals in the fish embryo. And so this again is ABCB4. When you knock it down, so we found when you knock down ABCB4, zebrafish embryos, so these are these arrows, they become more sensitive to the exposure to vinblastine. So vinblastine is a known cancer drug, and ABCB4 actually keeps it out of the fish embryos. So it becomes more sensitive when the transporter is knocked out or inhibited by a chemical. And accordingly, it also keeps out dyes, so calcium AM, for instance, or rhodamine. And we use that to look for the, to make or perform kind of a dye uptake assay with fish embryo, with live fish embryo, using the so-called VAS system. So this system is an automatic, semi-automatic system. It drags the embryo into a capillary and positions it in front of a lens of a microscope, and then fluorescence photographs can be taken from the embryo. And as you can see here, so when you don't add any inhibitor, the embryo is quite dim. But when you have an inhibitor of the transporter present, of this efflux transporter, the embryo appears much brighter, and you can quantify this signal, and then quantify kind of the effect of the inhibitor on the efflux transport. And in parallel, we performed ATPase assays with recombinant zebrafish ABCB4 protein. And well, the principle is that when a compound interacts with a transporter, ATPase activity is stimulated and phosphate is released from ATP cleavage, and you can quantify that, measure it. And we looked for correlation. So we tested a number of chemicals and looked for correlation of the results. So we performed single protein ATPase assays and whole embryo dye accumulation assays on the other side. And as you can see, there is a correlation of the data, but there are lots of outliers. And we think actually that this is a sign that there are other transporters in the zebrafish that also are responsible for efflux of rhodamine dye. And so other transporters are active. And we looked for another transporter when we had actually this data. So this is an image of zebrafish embryo, and it was exposed to the compound bromoxynil. So bromoxynil is a herbicide containing brome, and you can actually visualize the brome. So on the left, you see a laser ablation MS image, and it kind of detects the brome in the bromoxynil. And you can see here the a signal in the, it's kind of in the middle of the embryo, so probably the intestinal tract. And a similar image we got with a multi-MS image, where you also actually see the signal here in the middle. So it's probably the gastrointestinal tract. And the gastrointestinal tract expresses a certain protein, OATP1D1 is called. It's an SLC transporter that we studied, and it's OATP1D1 is an uptake transporter, and cells were transfected with this transporter. And when they were exposed to BSP, the ones that were transfected, so overexpressed OATP1D1, took up much more of the BSP than the control cells, which is kind of expected. It's an uptake transporter. But when the cells were exposed to bromoxynil, it was quite an interesting result, because there was less accumulation of bromoxynil in these cells than in the control cells. So, which is an indication that this transporter, which is supposed to be an uptake transporter, also has an efflux function. Yeah, and this, you know, when we got this picture, we thought, okay, it makes sense. It's an uptake transporter, so probably it so the cells of the gastrointestinal tract accumulate more bromoxynil, and therefore we get the signal. But when you think about it, okay, when it's an efflux transporter, it may also make sense, because it effluxes the bromoxynil into the lumen of the gastrointestinal tract, which might happen, yeah. So, okay, so basically the take-home messages, so transporters are important toxicokinetic relevant parameters in the zebrafish embryo, and we want to right now look at more ADME-related enzymes. So we go to human and look which pharmacokinetic or enzymes do we find, look for more candidates in the zebrafish. Are they expressed? What are the functional properties? And what is how are they involved in toxicokinetics? So the work that I showed was basically done by many people. So these were main main people like Stefan. He did a lot of the ABCB4 work. Katharina did the OATP1D1 research. Flavia actually worked with the VAST system, and Kathleen did all the ATPase assays. And these were the funding sources. Thanks very much for your attention.
And thank you for a very interesting presentation, Sil, and for sticking to the time. So, yeah. I mean, I have one question. So how many transporters are expressed in the zebrafish? Because you have all these gene expression data you mentioned, both RNA-seq and microarray.
Yeah, actually, you know, Paul is just doing the calculations, and I sent him a few. I really don't know how many, but it's quite a number. So he looked for different ABC transporters, and a lot of those, so these MDR transporters, so also ABCC, 1 till 6, I think, he looked at, and they pretty much all expressed. It's just you have to, because I said the expression is quite dynamic, so we have to really look at which stages. So at a certain point, they are all expressed, but in the very early stages, expression is quite low. So, but yeah, good question. But we really have to look further into that, what the expression patterns are.
Do you plan to knock them out like one after one, or what is your plan to dissect it?
That would be very great, yes. Yeah, I'm just starting this. So I did, Stefan was a PhD student many years ago, and I didn't do a lot of research now, but plan to really kick off now with zebrafish and do more work and use the CRISPR-Cas maybe for knockout and all that. So, but we haven't started yet right now.
Any other questions, Anna?
Yes, I would like to ask a question. We have seen that many detergents are substrates or alloprides for transporters, and at higher concentrations, they also inhibit the transporters. And now I wonder, I mean, we let a lot of detergents, tons of detergents per year into the water. And I'm always very much afraid that, you know, we come to a point where the water is so contaminated that these transporters may not defend their beings, the living, the fish and anything, the embryos particularly anymore. And do you see something, I mean, do you also feel that this is a danger, or what is your feeling about detergents?
Yeah, so it's always a question if those compounds reach the transporters. Yeah, so it's a matter of uptake and where they end up. So it's actually hard to say. So I tell you right at the beginning, I really don't know, but there is definitely concern. And also because there are many compounds. So as we heard already, now the transporters are multi-specific, and and we have thousands of compounds in the environment. And even if they are at lower concentrations, together they can act, in a combined way. So that is really a concern, and something I would like to address more. Yeah, because we have also some evidence that certain compounds, they act as inhibitors, as you mentioned, for detergents, but also other compounds like fragrances or certain pesticides also.
Anything which is amphiphilic, yeah.
Yeah.
Probably.
And yeah. So that is definitely concern we also have, that could be a significant effect. Yeah.
And the point is, even if they are not toxic, so that's the, detergents are per se not toxic at low concentrations at least, and but still they do the effect together with others.
Exactly. So
Thank
they
you
actually,
very much.
Oh, it's a valid point. Absolutely.
To add, yeah, you're right. So they are not toxic, and so they are not considered as toxicologically relevant. But this effect as, let's say, chemosensitizer is not not considered, at all in regulation. Yeah.
Okay, thanks a lot for a very interesting talk and discussion. So I wonder, should we try to put Dr. Chen's presentation on, or move on to Ian's presentation? Susan, what do you say?
I think Dr. Chen can start his presentation.
Okay, great.
Yeah.
So
thank you very much.
Oh,
And
yes.
also, I just want you to know that Lori received my PowerPoint, and she's going to help me to show the slides.
Dr.
Okay.
Chen, can you
Yeah.
see the screen?
Yes, I can see. Is that all the participants can see my slides? Okay, so I'll start. First, I would like to thank the Journal of Cancer Drug Resistance and Dr. Littman for organizing this meeting and inviting me to present the study we have done in the area of extracellular ATP induced and ABC transporter-mediated drug resistance in human cancer cells. Here, I would like to emphasize, first, we study extracellular ATP, not just ATP in general. And secondly, I have to admit I'm not an expert in the ABC transporter itself. It is our cancer study leads us to drug resistance. Next slide, please. Yeah, this is a slide shows the structure of ATP. Certainly, you all know ATP, so I don't want to talk much. But here, I would like to emphasize that ATP is not only a substrate, but also a cofactor in RNA synthesis. But at the bottom, I would like to mention two things. Extracellular ATP is a messenger for purinergic receptors located on the plasma membrane of animal cells, including cancer cells. Very importantly, ATP actually is a transcription cofactor. It's involved in initiation, elongation, and termination of transcription. So this is a factor actually plays a very important roles in our research. ATP is unique in that it is involved in biochemical reactions at transcription, translation, and the metabolism level. And ATP and ATP concentration are known to be closely involved in drug resistance in cancer. And first, I would like to point out that the cancer cells show higher intracellular ATP levels than normal cells from which cancer cells are derived. And even more interesting, that is the cancer-resistant, drug-resistant cancer cells actually show even higher intracellular levels than regular cancer cells from which the drug-resistant cancer cells are derived. So the intracellular ATP levels are correlated very closely with drug resistance status of cancer cells. Next slide, please. And solid malignant tumors are also known to have very high levels of extracellular ATP concentrations. Its concentration actually is 1,000 to 10,000 times higher than the ATP concentrations found in normal tissues. The normal ATP concentration are usually in the nanomolar range, whereas the extracellular concentration in tumors are in the 100 to several hundred micromolar range, as shown by this bioluminescence imaging of nude mice. On the right, certainly these are the tumor-bearing mice. On the left are healthy nude mice. Next slide, please. And so when my student, Maria Evers, added ATP to human non-small lung cancer A549 cells in migration and invasion assays done in transfer assays, as shown on the top right, she found that 0.5 millimolar ATP, that is the ATP concentration found in real cancer, real tumors. ATP induces significant more cell migration and invasion in vitro. In this experiment, the TGF-beta serve as controls in the study. As you can see on the rightmost pictures, that ATP induce more migration and invasion than even TGF-beta. Next slide, please. So using the same cell viability assay, in the cell viability assay shown on the left, my other former student, Sean Wang, found that EATP treatment reversed the cell proliferation inhibition exerted on A519 cells by target drug sunitinib. And the reversion is ATP dose dependent. She also did a clonogenic assay shown on the right, in which the drug sunitinib significantly reduced the viable clones in the middle of the 3 plates. But ATP rescued some of the sunitinib inhibited cells and formed significantly more clones, shown on the right. The bottom is the quantitation of this clonogenic assay. Next slide, please. So using the same cell viability assay, Sean went on to test more anti-cancer drugs, including both targeted and chemo drugs, shown on the top. She tested these drugs in 5 different cancer cell lines in 5 cancer types, such as lung, liver, breast, colon, and pancreas. This study shows that EATP induced the resistance to different drugs in different cancer cell lines. These results are shown by her other studies to be due in the fact that the different cancer cell lines express different ABC transporters. And different ABCs is certainly efflux of different drugs. And also, I would like to say that these plus signs means that the cell lines increase survival or drug resistance when treated with ATP. In a different study, in an ATP assay, my student also found that the EATP treatment upregulated intracellular levels of these cancer cell lines by 30 to more than 100% in the intracellular ATP levels. Next slide. So by observing these initial observations, we ask ourselves, what are the mechanisms of the EATP-induced drug resistance? Next slide. So in this slide, my student did an ATP internalization assay to determine if EATP can be internalized by cancer cells. So in this assay, she used a non-hydrolyzable green fluorescence ATP as shown in the middle. I'm sorry, I think you skipped a slide. Can you go back 1 slide? Oh, I'm sorry. Apparently, somehow that slide lost. Yeah. So this is an in vivo study. As you can see that we used a red fluorescence high molecular weight dextran, which is a macropinocytosis tracer in this study. And in the middle, we used a green non-hydrolyzable fluorescence ATP. And we co-injected these two molecules into the tumors grafted on the nude mice. And when we overlay the two photos on top of each other, as you can see, that the spots become yellowish, which means that these two molecules are located in the same cellular vesicles. And since the red fluorescence dextran is a macropinocytosis tracer, so we concluded that this indicates that the ATP can be internalized by macropinocytosis into not only cancer cells, but also in the tumors generated in vivo. And at the bottom, as you can see, certainly is a low magnification of the tumor cells. You can see a lot of internalized ATP, and they're located in the micropinosome. Next slide, please. And this is a study that Xuan used in an ATP assay. And in this assay, as you can see, the x-axis is time in minute, and the y-axis is relative intracellular levels, with untreated A519 cells being arbitrarily set at 100. The black circles, the bottom half, represent untreated control cells. Black square, solid square are ATP A519 cells treated with sunitinib, the drug, showing a slight decrease compared to the control. In contrast, the cells treated with ATP show a large increase, as you can see on the top curve, in intracellular ATP levels, while ATP treatment also significantly increased the intracellular ATP levels in cells that are treated with sunitinib. That's the second from top. Next slide. So in this study, another graduate student then used radioactive anti-cancer drug, gefitinib, and also paclitaxel to do a drug efflux experiment. In her study, A519 cells or H1298 cells, shown on the bottom, were incubated with these two drugs. After 1 hour, she removed radioactive drugs by replacing the incubation media with new media that contain with or without ATP. So after incubating cells with different amount of ATP for different times, she stopped the experiment and then measured the radioactivity inside the cell. These assays provide evidence that these cancer cells treated with ATP showed lower radioactivity inside cells, indicating that ATP promoted drug resistance by increasing drug efflux activity and pumped out the drugs. Next slide, please. Oh, apparently for some reason I lost some slides. Can you go back? So apparently you didn't receive all the slides. But then I'll just summarize after this. And going up, just keep. Yeah, just stay here. And Haiyun went on to do a Western blot analysis on the ABC transporter proteins from cells treated with ATP for various times and using TGF-beta as control. So the Western blot shows that ATP altered the level of ABC transporter proteins, but not by much. Therefore, we concluded that Eatp increased drug efflux primarily by increasing the intracellular ATP levels and secondarily by altering ABC protein levels. Unfortunately, I couldn't show you that Western blot for some reasons. And also, next slide. Next slide, please. And also, my student went on to do a metabolomics study on the ATP-treated A519 cells. And she generated a heat map, and unfortunately, I couldn't show. The heat map shows the all the cellular metabolites that are natural efflux substrate of different ABC transporters. And heat map shows that the level of the metabolites in general become lower over time of ATP treatment compared with the control. So these indicate that these natural metabolites are indeed pumped out, very likely through the ABC transporters. So here since I lost a few slides, I'll have time to read my summary. So Eatp-induced drug resistance to anti-cancer drugs in multiple cancer cell lines and multiple cancer types. Extracellular ATP induces ABC-mediated drug efflux by providing energy to ABC and altering ABC protein levels. Eatp is internalized by cancer cells by micropinocytosis. That should be a MA, not MI, macropinocytosis, leading to significantly elevated intracellular ATP. For example, in A519 cells, we commonly use the intracellular ATP level actually is increased more than 100% sometimes. So the elevated intracellular ATP drives ABC-mediated drug efflux by providing additional energy to ABC and by upregulating protein phosphorylations. I couldn't show you the model because we have other data that shows that the increased intracellular ATP level actually increased the protein phosphorylation, such as the cell membrane bound growth factor receptors that significantly increase the cell survival of the treated cells. And also, I don't have time to mention that Eatp also induced drug resistance by several other mechanisms, such as the induction of transcription and also the binding and activating of purinergic receptors. So can you go back one slide? Yeah, I would like to acknowledge the funding resources and also my students, including collaborators, and also my university, the department and the college, and also certainly the Journal of Cancer Drug Resistance for this presentation. So I would like to thank you very much for your attention, and I would like to answer your questions if you have any.
So thank you very much for a very interesting presentation on the role of extracellular ATP on drug resistance. Actually, it reminded me of my first year at NIH in 1997, where I was at the basement together with Ted Abraham, who thought P-glycoprotein was an ATP efflux transporter, which we couldn't show though. I think it's interesting data. I didn't see a dose response where you increase the concentration of ATP. So I think you would have to show that to substantiate the claim that there is a dose response effect. Also, I didn't see any levels of ABC transporters, but maybe you have data that are not shown. And as regards the NHF ATP, I mean, that could be non-transported because it is NHF, not hydrolyzable ATP. So it could explain the transport you see. I think it's very interesting. And of course, your observations that ATP is high in cancer is absolutely real. I mean, we know that could even be explored to treat cancer if you could deplete the mitochondrial ATP production. As regards the link to the ABC transporters, I think that remains to be seen because you would need to show a dose response, in my opinion.
Actually, we have those data showing our publications, and but unfortunately, because of the time limit, and also I lost a few slides. Actually, I do have Western blot, and also we do have plenty data that shows that the extracellular ATP significantly induce intracellular ATP levels, and also we know that when the extracellular ATP is internalized by micropinocytosis, and it is by the function of a micropinosome, and micropinosome actually is leaky, shown by others, and so the ATP could be released into intracellular environment, and then functions both as transcription factors. Actually, we did a RNA sequencing analysis that shows that the ATP, actually extracellular ATP, significantly upregulated many drug-resistant genes, including ABC transporters.
Yeah, I would like to see that. Of course, you have pure energy receptors and extracellular ATPases as well, so that could explain it. Anna, do you have a comment, a question?
I have a question. I didn't quite get the micro or the pinocytosis. Is it working in healthy cells and in cancer cells, or is there a difference between the cells?
Yes. First, I would like to mention that macropinocytosis currently is considered to be one of the hallmark of cancer metabolism. That is, macropinocytosis is significantly upregulated in cancer cells, but in normal cells, such as some of the immune cells, actually, they also use macropinocytosis. But I did not have time to mention that. So the ATP actually not only is internalized by macropinocytosis, but also by several other endocytosis processes.
So do you know whether it's correlated with the expression level of of transporters? Because immune cells also may have high expression levels of transporters.
Yes.
Pinocytosis.
Yes. I did not study immune cells. I just studied cancer cells. In all the cancer cells, actually, we found that macropinocytosis are primarily upregulated very significantly. But one thing I would like to mention that there's a famous mutation in cancer cells called KRAS. And in KRAS mutated cancer cells, one of the functions of KRAS mutation actually is upregulation of macropinocytosis.
Okay, thank you very much.
Yeah, thanks a lot again for interesting talk and discussion. In the interest of time, we need to move forward to Ian, if you're ready with your presentation.
Slide should be coming up now, Thomas.
Yeah, looks good. All yours.
And it should have moved to presenter mode, but whether it has or not is another matter. Is it on presenter mode?
It's fine, whatever works.
It's fine, I'll carry on, as long as people can see the slides as I move between them. So thanks very much for the invitation to talk this afternoon, Thomas. It's much appreciated. And what I want to tell people is a smaller part of a much bigger story. So I've had a postdoc in my lab, James, for a few years now working on a BPSRC grant. And I just want to tell you one bit of his work, which is trying to develop an assay using time-resolved FRET to explore inhibitor and substrate binding to the multi-drug pump ABCG2. As we've heard, ABCG2 is a broad substrate specificity transporter. It's localized to many so-called sanctuary sites in the body. In terms of cancer drug resistance, we know it is clinically relevant, especially in leukemia, but there are some solid tumors where expression of ABCG2 is a negative prognostic indicator. We know also that due to its localization in things like the gut and the kidney, that it affects the pharmacokinetic profiles of many drugs. And slightly different to ABCG1 that we've heard quite a bit about, we know that G2 has to function as a dimer because it's a so-called half transporter. So in terms of drug binding and transport, we know that G2 is polyspecific, and I've just listed here a handful of chemicals or types of chemicals that we know that ABCG2 can interact with. And Anna knows probably thousands more that it can interact with and how. Those range from physiological substrates, which include urate and probably include some heme-based compounds, to clearly non-physiological pharmacotherapeutic compounds, such as statins and anti-cancer drugs. And we know, as Michael has said earlier, that there's some overlap with the substrate repertoire of another multi-drug pump, PGP ABCG1. And we're interested in this polyspecificity. And there are many ways in which people have investigated this. And these includes looking at the transport mediated by ABCG2, either in cells or in inside-out vesicles, or measuring the transport across polarized cells, or looking at the ability to stimulate the ATPase assay, to stimulate the ATPase activity in membrane preparations, or to just see if you can solve the crystal or the cryo-EM structure with your favorite drug bound to the protein. And there's been a lot of advances in that for ABCG2. And then there's also a whole range of mutational analysis. Can we somehow perturb the pump and show that that affects drug binding and transport? So our aim, one of our aims in this grant, was to see if we could develop a technique that was purely focused on the first step in translocation. In other words, could we get a technique working that looked at how drugs interact with the transporter? And the assay we've been working on is a FRET-based assay, which is shown schematically on this slide. So what you're looking at this slide, hopefully my pointer is showing for you, is in both cases we have one of the ABCG2 structures. And on the left-hand side, we have this orange circle. This is a fluorescent molecule bound in ABCG2's so-called cavity 1. And this fluorophore is accepting resonance energy from a terbium atom. And that resonance energy transfer can be detected to show that that a fluorescent molecule binds to ABCG2. And I'll explain more how we get the terbium atom near our fluorescent compound in a minute. On the right-hand side, we show what happens if you have the exact same setup, but in addition to this fluorescent compound that binds to G2, you've also had another non-fluorescent compound that would displace this molecule. And in that case, we no longer get a FRET between the terbium and the fluorescent ABCG2 substrate or inhibitor. So this is the principle of our assay, to use the light emitted from excitation of terbium atoms to measure fluorescence binding at ABCG2. And the way we've tagged our protein with terbium is to take advantage of a protein tag known as the SNAP tag. Now, the SNAP tag is a smallish 18 kilodalton protein. It's an alkyl guanine DNA alkyl transferase protein. This means it can bind covalently with anything that is that has an O6 alkyl guanine. And so we've tagged ABCG2 with this so-called SNAP tag. This protein is then labeled with an Oregon green version of O6 alkyl guanine, and that lets us show that our SNAP tagged ABCG2 is present at the cell surface. On the right-hand side of this slide, we can also show that this SNAP tag G2 is still functional. So this is a conventional transport assay looking at the accumulation of mitoxantrone. It's probably just worthwhile looking at the bottom half of this panel. So when we add mitoxantrone to cells expressing this SNAP tagged ABCG2, then they do not accumulate much mitoxantrone. That's this population, this fluorescence of mitoxantrone on this axis. When we add the classic inhibitor of ABCG2, which is KO143, we shift this population to the right, which shows that they're now accumulating mitoxantrone. So this SNAP tagged version of the transporter is at the cell surface and it's functional in a transport assay. And what we then designed and made was a version of KO143 bound to a BODIPI fluorophore. So KO143 is a specific inhibitor, or reasonably specific inhibitor to be correct for Michael's benefit, of ABCG2. And when we add this BODIPI fluorophore to it, it still retains that inhibition, which is shown on the right-hand side of this slide. We then undertook these this TR-FRET assay using this BODIPI BY630 KO143, so this fluorescent inhibitor of ABCG2. And we could show specific and saturable binding of BODIPI KO143 to ABCG2 with an affinity of about 50 nanomolar, which is very, very similar to the sort of concentrations of KO143 that inhibit ABCG2 in cell and membrane assays. I should say all of these assays are done in cell membrane. We have extended this. We've got a whole range of mutants of ABCG2 where we've made changes in the drug binding pocket, changes in the pocket that should interfere with KO143 binding. And that data is not quite finished. That's why it's not on this slide, but it does seem to show that where we mutate residues that should interact with KO143, we do perturb this saturation binding isotherm, and we do see changes in affinity. What we've also done is we've shown that this fluorescent inhibitor, it does get displaced, so it doesn't just bind to G2 and sit there. It can be displaced in a sort of homologous fashion by KO143. So this is now the FRET ratio plotted against the concentration of the displacing ligand, and you can see that KO143 can displace its fluorescent counterpart with an IC50, again in a sensible range for this inhibitor, about 100 nanomolar. So we can use this fluorescent KO143 and this TR-FRET assay to measure specific binding, and we can measure competition as well with a high affinity G2 inhibitor. We can also start to measure substrate interaction with the transporter by looking at substrate's ability to displace the BODIPI KO143. We've looked at 3 substrates. I'm going to just zoom my screen a little bit because otherwise I can't see my own data. To show here that we can displace our fluorescent KO143 both with estrone sulfate and with imatinib. We can't displace it with a couple of compounds that we don't think G2 interacts with. There's no evidence in the literature that either aspirin or allopurinol interact with ABCG2, and we don't find that they're able to displace our fluorescent KO143, which affords us some confidence. What we are seeing is an ABCG2 specific phenomenon. And so I'm happy to wrap up there and say that this TR-FRET assay looks as though it allows us to measure binding as opposed to transport at ABCG2, and allows us to work with both wild type and mutant proteins and start to look at a wider range of substrates and other ABCG2 inhibitors. I think I've kept pretty much to time there, and I'm happy to answer a couple of questions if there are any.
Thanks. Eladia, very interesting and very clear presentation, and to the point and to the time. So it makes a lot of sense to do this FRET assay. How large is the terbium atom? I mean, does it interfere with the structure of ABCG2 or?
I mean, it shouldn't do. The snap tag is at the N terminus of ABCG2. There is a short link up before the rest of the MBD. Yeah. And so what we don't know is exactly where that snap tag is spatially. But you know, it's hanging off the end of the MBD. It shouldn't be interfering with anything going on in the membrane.
All right. All right. And do you plan to make any ATPase mutants also?
So we have got the classic Walker B mutation E211Q in ABCG2, and we have the data for that is predicted. Sorry, our prediction from that is that it would shift this curve. It would produce a shift in the affinity. It would reduce the affinity, I suspect. We haven't done the experiments yet, Thomas. One of the downsides of this is that the chemists were unable to make a massive amount of this compound for us. And so we've had this awkward task of prioritizing the experiments. This is one of the next ones to do.
Right. Okay. All sounds good. Good luck. Oh, I have a question for Michael.
Yes. In a very interesting and really very nice assay. One of the peculiarities of P glycoprotein is that substrate the residues in the substrate binding domains that should be interacting with substrates, you can mutate them and not see very dramatic effects on transport. It's almost as if there are primary and secondary and tertiary and quaternary binding sites. So you can substitute lots of alanines in the substrate binding site. This is work that Suresh Ambudkar has done. Do you know if the same is true in ABCG2? Have you looked at your mutants which affect substrate binding by your assay to determine whether they actually are affecting transport?
So one of the things I think literature tells us is that most of the multi-drug pumps have the potential to make lots and lots of weak but accumulating interactions with their transport substrates. And so that single point mutations may sometimes not have a big impact because there's another residue nearby that sort of then mops up and vowels interaction. So I think single point mutations are a difficult with any assay. Some of the mutations I mentioned in passing are not single point mutations. They're multiple mutations at different points in the pocket where we hope to be able to see a bigger impact.
All right. So thanks.
May I ask a question?
Yeah, sure. Anna, go ahead.
I've just been
Sorry,
question.
I would like to ask you for the conditions you measured did you have, what is the ATP and the magnesium concentration?
So this is all done in membranes that we anticipate are ATP free, Anna. So one of the things we want to do is also run this with AMP PNP and trap transporter in that ATP bound state.
This would be very interesting because this is probably the assumption that you have an open conformation when you have no ATP, and then you have the binding in this open A shape, and you have probably, so it would be very
Absolutely.
interesting to do it in the presence of nucleotides.
Yeah, we're very excited to do those experiments. Yes. Like
very
some of
interesting.
the data on here was obtained yesterday, so we're quite. If you call me in a week's time, we might have some of that data.
Very good. I'll call you. Thank you very much.
So thanks again for a great presentation and discussion.
Thanks, Thomas.
I don't know, Tito, are you still with us, or did you have to go to clinic?
Yes, I'm with you. Here I am,
Okay.
Thomas. I'll even start my video, and I'm going to share my screen.
Thanks a lot. And sorry about the delay.
It's okay. No problem. Can you see my presentation?
Yes, on the left side.
Okay.
Maybe you want to maximize it a little bit.
Yeah. There.
That's good.
Let me get you all out of the way. You're in my way here. I can't. Okay, and then I'm going to. How's that?
Yeah, that's fine. Perfect.
Okay, so I'll, Thomas, at the beginning in your presentation, you were alluding to, and some of the other titles sort of alluded to the fact that it's a shame that this hasn't gotten the clinical traction that many of us think it should have gotten. And I'm going to tell you why I think that's important. So nowadays, a patient presents to a U.S. oncologist, and the first thing that the oncologist, if this patient has either never been treated or has already been treated and the cancer has either not responded or has recurred, what is the oncologist most likely to say, or what are colleagues most likely to ask them? They'll send it to Foundation, and I'll explain what that is in a second. Or sometimes now we send it to CARES or send it to Tempus. Foundation One is basically an organization, a for-profit business that will sequence your DNA from the tumor that your patient has. Actually, what it does is it doesn't sequence the entire genome. It's not whole genome sequencing. It takes over 300 genes. It's actually currently 309 genes, 309 genes that we all think are important in cancer. And then additionally, 36 genes that are involved in rearrangements, and I'll come to that later, and basically gives you a report back on that. What are those 309 genes? They're shown here, and there'll be a quiz on this at the end of the talk. No, I'm just trying to point out to you, by the time you have 309 things on a slide, they get to be very small. But they're the kind of genes that we all sort of associate with cancer. P53 is here, KRAS is here, BCL2 is here, AXL is here, BRCA is here. So genes that we associate with cancer are in here. And then some years ago, after it had been started, they realized that, well, not all genes that are involved in cancer need necessarily mutated. In some cases, we'll have rearrangements that occur. So there are a set of 30 some odd genes, 35, that they look for rearrangements with the promoter of another gene and the critical portion of that gene. And in this context, I'll point out NTRK1 and NTRK2 here, which are genes that get sequenced in everybody and which get assayed in everybody. And they occur overall in about 1% of tumors. So that's how deep we're diving into this. We're willing to spend time, money, effort looking for a gene that is responsible for some role in only less than 1% of cancers. The output from that is that it basically classified mutations as category 1, 2, or 3. And basically, category 1 are mutations that we know are important in cancer and for which we have a drug. And ideally, for which we have a drug that has been shown to work in your cancer. Because, for example, if you have mutation in the EGFR and the epidermal growth factor receptor, and you have lung cancer, we've got really good drugs for tumors that have that mutation and have lung cancer. If you see that same mutation in, for example, a gastric cancer or a colon cancer, those drugs don't work. So there's something about the mutation and the tumor in which it occurs. So category 1 are those. Category 2 are, well, this might be important, but we don't have a drug. And category 3 is really, I don't know. It might be potentially important, but we don't have enough information yet. So what it basically does, it gives you a report. And what you see here, for example, and you'll see there that it says ALK rearrangements, which is something, again, that we see in lung cancer, and that's where they're important, not anywhere else. Although the ALK stands for anaplastic lymphoma kinase, where it's really been shown to be important clinically, is actually in non-small cell lung cancer. And there's 4 drugs that are currently approved for the treatment of ALK, and it gives you those 4 drugs as choices. And so they have a long list according to the cancer. Here's non-small cell lung cancer, melanoma, breast cancer, colorectal cancer, ovarian cancer, cholangiocarcinoma, prostate cancer, and solid tumors in general. And so they'll give you recommendations. But what you need to know is that the recommendations that they give exceed that for which they have FDA approval. And you need to know that the doctors use this, although there is really no FDA verification that this is important. So what's the problem? In the United States in 2022, it is expected that 609,360 Americans will die of cancer. And it's not because their tumors did not have, quote, actionable mutations, meaning mutations for which we have drugs, but because their tumors are, in fact, drug resistance. Everybody who dies of cancer dies with a drug-resistant tumor. This is a list that I stole. So this is the list of the genes in Foundation that are evaluated, and there's very few of the genes that we consider to be drug resistance gene. This is a list that I stole in part from Michael Gottesman, and it's a list of genes that confer drug resistance. Only a handful of these are found in the Foundation 1 list. The reason is that they primarily confer drug resistance not by acquiring mutations, but by overexpression or underexpression. Looking for mutations in these genes is unlikely to provide answers that let us understand why tumors fail to respond and why patients die. So we know that it's expression. So since this is about ABC transporters, Susan and I worked on this long ago, and this was in the mid-1990s. What we did, we looked at the expression of genes in the NCI anti-cancer drug screen, which was 60 cell lines. And basically the way the data was plotted at that time, and still is you would get for any drug, basically a profile that would show you how active it was among the 60 cell lines. And the central point was the median of all that activity. So you were either to the right or the left of that median. What we did was we looked at the expression of PGP in cell lines, and we were able to correlate lack of activity, shown here as these bars coming over to the left, with high levels of expression of MDR1. In other words, expression of drug-resistant genes was what mediated drug resistance. We identified in this way nearly 500 substrates amongst those NCI cancer drug screen drugs that have been screened in the NCI cancer drug screen, and the majority of those were indeed P-glycoprotein substrates. So then why do we do this? Why do we send our list of Foundation if we're not getting all the information, and indeed what I would argue the important information? The reason is because this is difficult. So what you here see is an expression array. It's difficult to interpret. It's much easier to look for mutations than to look for changes in expression. And then that's why we end up doing this. So what about the rearrangements? Well, we look for those, but again, we're not looking for rearrangements that confer resistance. And in ovarian cancer, this large group of individuals not long ago reported that they had found the rearrangements involving P-glycoprotein and a variety of genes in ovarian cancer. Therein lies the problem. The genes that were linking to P-glycoprotein and upregulated it varied across the different cancers. And searching for this would be very difficult. Foundation looks for rearrangements that are very well defined. By the way, in this study, which I was finally alerted to by Pete Borse, who wrote me an email one morning, and all that it said was finally, and I knew what he was talking about. They talked about gene rearrangements as the cause of resistance. They failed to overlook the fact that 15 years earlier, we had reported this, and that had they done a simple Google search for MDR1 rearrangement, at the top of that Google search out of 102,000 results would have been our publications. We were able to show this even in samples from patients with leukemia. These were children with leukemia, and what you see here is probes that were looking at the rearranged genes, both the P-glycoprotein and the now important promoter of another gene that was upregulating the expression. So none of these individuals failed to find that in the literature. And it always reminds me of what Dan Sackett, who used to work with us, and we wish he still did, but we now at Columbia and he's at the NIH, used to say, an hour at the library can save you a year in the laboratory. So is this important? In fact, it is. So Dr. Bates and I have analyzed the growth and regression of clinical data from over 140,000 patients. And what you can do is you can take the actual growth and regression of tumors, which is what you see normally when you're treating cancers, an initial regression and then a subsequent growth. And you can basically break that down into a fraction of tumor that is regressing because it's sensitive to the drug, and a fraction of tumor that is growing because it's resistant to the drug. There's absolutely no correlation between regression of the tumor and the patient's overall survival. But growth of the drug-resistant tumor is what impacts the patient's overall survival. And we can do this because when we look at the rate of death and we compare it to the rate of growth, we can see across all cancers that the rate of growth, which we designate G, is indeed an excellent biomarker of overall survival. What you're seeing here is we took over 800 patients with non-small lung cancer. We determined the rate of growth of their tumor, and then we looked to see what the rate of overall survival was. This is the best overall survival. This is the worst. This was the quartile of the data with the slowest rate of growth, and this was the quartile with the fastest rate of growth. This is true across all cancers, across 140,000 patients. So it is growth of the drug-resistant tumor that is responsible for why patients die. And unfortunately, we have no insight into what those genes are in any patients and whether or not their tumor have that, and we should be aware. So in summary, we test extensively, looking for what alterations in a tumor might lead to a therapeutic response. But patients eventually die of drug-resistant tumors. And in the overwhelming majority, that drug-resistant clone was likely there to begin with. It just grew out over time. It would be nice if we knew that. Eventually, having that knowledge will help us be better doctors, but that will be much more difficult than looking for simple alterations such as mutations, for which you can do this quickly and without a whole lot of problem and charge a lot of money. We will need expression data and knowledge of what level of expression is low enough to overcome and that we can ignore, and what level of expression is high and will prove lethal because it will prevent drugs from working. We've done the easy part, look for mutations. Now we need to think how we do the hard part. So with that, I'll end my slide. I'll stop sharing, and I'll be happy to take any questions.
Thanks a lot, Tito, for a very thought-provoking presentation and worrisome. I mean, so what to do if we're not testing? I mean, the test should be available. I mean,
Right. I mean, you know how difficult it is to express, to look at, cDNA arrays and that data, and interpret them, right? And then the problem is what we always know in P-glycoprotein, we always say, is that level of expression important? Is it significant to confer resistance? So we have to come up with values for that. And we probably would have to have values for different cancer, because possibly a little bit of P-glycoprotein in one tumor might be enough to confer resistance, but in another tumor, you may need larger amounts. And you may need larger amounts for some drugs than you do for others. It's a complicated thing, In the laboratory, we can do this, but we've got to figure out how we do that with patients.
Right. And it could be a double-edged sword, as Michael might comment with his question. Michael?
So, no, first of all, Tito, thank you for helping us think really important thoughts about the issue of treating drug-resistant cancers. You probably know that one of the things that we're doing now in my lab is we're trying to create a larger catalog of genes that are important in drug resistance. We know the ABC transporters are, but there are clearly, as you pointed out, many, many others. Do you think it's possible? I mean, I think we're doing these CRISPR analyses. We activate, we inactivate, we knock out genes. We're getting a lot of genes that get in tissue culture with resistance to specific anti-cancer drugs. It's going to be hundreds. It's probably going to be many more resistance genes, and we know that already, than there are oncogenes. Is this a solvable problem? Is it going to depend on there being major players and minor players, and so you can deal with most drug-resistant cancers, but not all? Or can we actually figure out how to solve this problem?
I mean, I think we can figure it out. But then the question is, will people be interested in what we figured out?
They haven't been so far, I guess.
Right. Yeah. So what happens is, you get an EGFR mutation and, oh yeah, we got a drug for that can be used and that a company can sell it, right? So it would have to be in a situation where you have more than one option, which is increasingly common. Okay. And I have drug A, B, and C. And if a mechanism of resistance that you identify is going to impact drug A, that then becomes important because you have B and C as a choice, and you won't use A and you'll use B and C. So I think that, yes, we're heading in that direction because we have an increasing number of options for cancers. And you sit there and you say, which one am I going to use? I'm going to throw A or B or C, like in prostate cancer. There's a plethora of options now in first line and second line and third line. We need guidance. And the majority of these aren't mutation directed, but they clearly are mechanisms of resistance. And if they can be identified, then it can inform a doctor's decision.
So one big advance, I think, is that the FDA now requires that when a new drug comes on the market, that there be some description of how it's handled metabolically and by transporters like P-glycoprotein. So that's sort of encouraging some of the companies who are making anti-cancer drugs to shy away from known PGP substrates or known G2 substrates. What I'm wondering, though, is whether or not and then maybe a great example of that is you mentioned ALK positive non-small cell lung cancer. That's maybe 5% of lung cancer. And the original drug, ceritinib, which was used to treat it, is an incredibly good PGP substrate. And when you look at the tumors, a lot of them express high levels of PGP when they're resistant. But the second and third generation drugs are not very good PGP substrates. So that maybe that's an example which took industry and oncologists a long time to learn that this is something pretty much figured out up front and saved a lot of development time. Maybe not made as much money. That's the problem.
Right, exactly. But no, Michael, that's a perfect example because they're still out there. And maybe you don't tolerate B and C. And you have to go back to A, the original one. So, which was the one that was a strong PGP substrate. But if you knew that the patient's tumor doesn't have PGP, okay, and then you also know that they don't have brain metastasis because that's where the non-PGP substrates were actually more effective in getting into the brain and treating the brain metastases that were there. But if you knew that, both of those things, then if A is tolerable, you have an option. But So yeah, there's already examples of where knowing that would have been of value. And so what now needs is a little bit of financial incentive to drive that. And maybe as a community, what we ought to do is say, okay, which are the ones that are, if we know that they're drug resistance genes, that they're going to confer resistance, and look, forget whether the level is 10, 20, or 100. All of those are bad. So that we don't have to factor in that, is this level important or not? Just any level is important, okay? And try to push forward a few of those as genes that we should start to be considering. So we ought to talk more, Michael. We ought to think about what drugs are available, where our choices are available, and do we know some drug resistance genes that would inform the decision? I think we can come up with some good suggestions. And then one has to conduct an experimental trial, maybe even in the real world, where you'll check for expression of that in the tumor or not, and let that inform your drug decision and see how well that does.
There's another element. You mentioned that it's likely that many of these mechanisms of resistance pre-exist in heterogeneous tumor populations at the time of presentation. But let's say a fair amount of resistance evolves during treatment. We need a way of, and you know that physicians and IRBs are very reluctant to allow you to continuously biopsy solid tumors during the course of treatment.
Right.
So you do need a way of detecting maybe circulating.
The emergence. Yeah, and also, right. But, circulating, they're doing that now for DNA. But boy, circulating RNA, that's going to be really challenging.
Oh, because it doesn't.
But by the way, IRBs and all these people, but there's a lot of patients that are very motivated. If you tell them, if I biopsy your tumor, I might get meaningful information, they'll say, do it. Sign me up. Can you do it right now?
Yep.
Or at the latest tomorrow? So I think, we just have to we have to think hard about it and see where we, model this, where we bring it to people and say, look, this is meaningful. Let's see. So I'll reach out to you.
Yeah, let's talk.
Okay. All right.
So that was great.
Okay.
Having you guys discuss this very important topic. And I hope you will continue the discussion. Now, I think, Susan or Laurie, you might have something to say on the journal, because I think this was the free discussion. We don't have time for more free discussion, actually. Yeah, 40 minutes over time. So please feel free to give your journal introduction, please.
Okay.
Or do we have the vote now?
I think we should do the vote and then the journal introduction. Is that okay?
Mm-hmm. So you are in charge of that.
Okay, I will release the. Can you see the poll of best presentation award?
Yes.
Yeah. And we have 30 seconds to finish the poll.
If I'm supposed to be seeing something that I'm supposed to do, I'm not seeing anything that I'm supposed to do.
There was a pop-up screen where you could put a mark on. Now it's gone.
Okay, it's all right.
I think there should be, Tito, you might see along the bottom a button next to the share screen that says poll/quizzes. If you click on that, then it will come up.
I see that.
Hello, all experts. I have received the results, but there are similar votes for 3 experts. So we need to do it again, and I need some time to prepare the poll of. I need some time to prepare the vote. So Laurie will do the presentation first. Okay.
Okay.
Hi, this is Laurie. Can you see my screen?
Mm-hmm.
Okay, now I'm going to introduce the CDR journal. And the editor-in-chief is Godfrey Peters. And this is the editorial department team. Susan Song is responsible for journal management. Laurie Kahn mainly take charge of special issue construction, interview, and the webinar. Blanda Zhang is responsible for manuscript processing, and Bella Zhao take charge of conferences, cooperation, and promotion. So if you have any questions, please contact any of them to solve your problems. CDR is an international peer-reviewed open access online journal. And the journal publishes research articles, reviews, case reports, commentary, and letters on aspects of drug resistance and its reversal, including drug design, drug delivery, drug distribution, and ancillary drug resistance. And these are some data of the cancer drug resistance. And our company cooperates with some professional institution. And at the right side, we can see we also cooperate with many international conferences, which could increase the exposure of the cancer drug resistance. And up to now, we have indexed by following database, especially in May, CDR is officially indexed by PubMed, and in August, CDR is officially indexed by ESCI. Next, I will introduce the editorial board distribution. We can say there are 67 editorial board members from 17 countries, most of them from United States, the Netherlands, Italy, UK, Germany, China, and other European countries. And each index from 40 to 60. So such an editorial board member could help us achieve a higher level. And our junior editorial board member also do a lot of support for CDR development. With the effort of all editorial board member and editorial department team, we strictly control the quality of the articles. So we listed some of the high cited articles published from 2020 to 2022. The data from the Google Scholar. In order to get our journal more better, we do some innovation. We can see here is a homepage, and at the bottom of the homepage, there are lots of data, and the homepage and this data is updated in time. And for article page, it's easier to download PDF version here, and we also can see the data of the downloads and the viewers. We also encourage all the authors to provide a geography abstract, which could increase the exposure of the article. And for webinars this year, from September we start to hold online webinars to enhance interaction and exchange among scholars. So for all of the experts, if you are interested in holding the webinars, please feel free to contact us, and we will help you to hold all of the procedure. Except the webinars, we do a lot of interviews this year. From July to September, we conducted 4 expert interviews. So we will continue to conduct interview with experts to strengthen the cooperation between expert and editorial team. At the same time, spread their thoughts to the world. So, so if you have any current research outcomes or publish a new article, we are happy to help expert to spread their thoughts and do an official interview. In order to get more exposure, and we also establish a lot of official account, like Twitter, YouTube. So please follow us and contact us. Lastly, exciting news. The 2020 Best Paper Awards has already been set, and will announce at the end of the January 2023. We will launch the 2021 and 2022 Best Paper Awards successively. Lastly, thank you all for your presentation and your precious time. And lastly, I want to ask a question for our expert. So would you mind us publishing your presentation on our official website or on our YouTube website for promotion? Hi, Thomas. Hello. Sorry, I can't hear you.
Yeah, now I'm unmuted. Yes.
Would you mind us publishing the presentation today?
No, I'm fine.
Okay, thank you. So other experts?
Yeah, I have a problem because we had some unpublished data in the presentation.
Okay. So if some expert have some data cannot be exported, we will do not publish it on our official website.
Okay.
I can publish it.
Okay.
Thank you.
So are we back to the poll now?
Okay, according to the poll, Dr. Michelle, Dr. Ian, and Dr. Chen got the same numbers of votes. So we need to vote again for them to have the final winner. I will launch the poll again.
Did you launch it already? I don't see it. Oh, here it is.
Okay. Okay, the poll is finished now, and I sent the winner to Dr. Thomas. Can you see in the, yeah?
Yes.
You can announce
So
the winner.
I will announce the winner, and I'm happy to announce that Dr. Ian won the prize for the best presentation today. Congratulations, Ian, in absentia, because I don't think you are, yeah. So, yeah.
Okay, congratulations, Dr. I. And after the meeting, I will contact you about the related information about that.
Great.
Okay.
Excellent.
Okay, I think our webinar can be finished since it has almost 3 hours. Yeah.
Yeah. So I just have a concluding remark. That's again, thank you to all participants, especially all those who stayed all the time. Thanks, Michael, Anna, and friends from abroad for joining us. And I think it was a very interesting discussion. I hope we can continue it offline. So thanks a lot. Take care.
Thanks a lot, Thomas.
Thank you so much.
Yeah. Great seeing you. Bye.
Bye.
Thank you. Bye.
Thank you.
Bye.
Bye, Anna. Anna, here.
Bye. I have a Wackelkontakt in German. My camera is, sometimes it works, sometimes it does not work when I shift it. I'm sorry. Bye-bye. Thank you, everybody.
Thank you.
Bye.
Bye.